genetic point of view.
Interesting. Thank you. Dr. Morimoto.
Yet another wonderfully interesting question, Laura. If you think about each of us humans, we have 3,000 to 5,000 single nucleotide polymorphisms. Within the 20,500 open reading frames that make proteins, we know that there's between 9 and 13,000 missense mutations that distinguish the human population. Missense mutations never make proteins better. They can interfere, and what's interesting is, then when the quality control machinery changes in aging, now these missense mutations that were once benign in youth now become terrifying. And so they add, I think, remarkable complexity to how the system then tries to maintain balance, and why, when you think about all of the age-associated diseases, they don't all flip at the same moment. Okay, they happen over a large span of time, depending on genetic background.
Very interesting. So could you distinguish between proteostasis sort of within the mitochondria versus outside of the mitochondria, and which is most dangerous?
Mitochondria are so central because every step in protein quality control requires an ATP for a peptide bond and an ATP for translocation and an ATP for degradation. And so the mitochondria is really at the epicenter. When the mitochondria dysfunctions, remember that 99.5% of the proteins in the mitochondria are encoded in the nucleus, have to be synthesized in the cytoplasm and transported. So an imbalance in mitochondria literally sends proteins back into the cytoplasm, where they're now overwhelming the protein degradative machinery.
Mm-hmm. Interesting. So Dr. Valenzano.
Yeah, I really much enjoying the conversation, and I agree with all that's been said. And just one thing I want to add is that if dysregulation is a sort of like a hallmark, if you wish, of the aging process, then heterogeneity is a sort of like is a consequence of dysregulation. Heterogeneity at individual level, at the level of tissues or cells or molecules within an organism, but also between organisms, to provide you like with a personal angle to this. We study, for example, the microbiome in aging. So we study how my microbiome is different from your microbiome. And if you look at microbial diversity or function in young organisms, this don't have to be humans, but also, for example, in simpler fish, for example, the microbiome of younger fish is much more similar in terms of structure, composition, and function than the microbiome of older individuals. In other words, every single individual has its own private sub-sample drifted microbiome, which leads on a kind of like broad statistics to increase heterogeneity in microbial composition, microbial diversity. So this is something that we observe over and over, this increased heterogeneity also at the level of microbiome. And also to provide you like one more aspect regarding sex differences in heterogeneity. In the organisms that we study, these short-lived killifish that live in ephemeral ponds in Africa, what we observe when we look at stem cells, hematopoietic niches in the main hematopoietic organ, there are massive differences during aging in actually DNA damage and genomic instability in different classes of progenitor cells, whether you are male or female during aging. So very different signatures that can be completely accounted for by the sex of the organism.
Interesting. So across the different species that you study, do you see a pattern that correlates with what happens with humans, would you say?
Well, patterns in increased heterogeneity for sure.
Yeah.
Whether the classes of cells that get dysregulated with age, or whether if this is what you're referring to, actually yes, we see very similar signatures of, for example, immune system, age-dependent changes occurring already at the level of these organisms with age.
Yeah. I can't help but ask because we study mice in my laboratory, and I think the environment is an incredibly important component, and we neglect it with our mice. How do you handle that with killifish or other model systems?
We try to maintain, we try to sort of, on the one hand, control the environmental variability by having
Right.
laboratory conditions which are as homogeneous as possible. Therefore, the differences we observe, for example, in male, female, aged fish, cannot be completely accounted for by environmental differences. At the same time, we try to also embrace environmental diversity in nature, for example, to understand how variation in heterogeneity across different individuals can be actually explained by environmental changes. Like, just to briefly comment on that. So, for example, we can study whether microbes, because microbes is a very important sort of like interface between the host and the environment. It's a very important layer of interface, like this permeable, if you wish, membrane between the environment and the host. So we can study whether heterogeneity in males or females can be accounted for by microbial differences, can be explained causally by microbial differences present in the intestines of either males or females. So we try to assign actually how the environment translates into the host and host-specific differences.
That's great. Yes, please.
So the other model system we work in our laboratories, an animal called C. elegans, which is a remarkable 959-celled animal. It's isogenic, so there's no genetic variation, which offers tremendous opportunities to do certain kinds of experiments. What we discovered is at 30 degrees centigrade, proteins are all folded, functional. At 34, a number of them are aggregated and amyloid. At 32 degrees, you now see inter-individual variation. Some animals, the proteins are crashing, and the same proteins in other animals are perfectly soluble. So it's directly to your point that rather simple change in environment, one that we can control precisely, has profound consequences.
It's a very useful system and quick to study. So
very inexpensive, too.
I'm jealous. Dr. Valenzano.
I wanted
Yes.
to comment briefly also on this. We actually studied this super long-lived vertebrate called the Greenland shark. They live for 180 years, plus or minus 120 years. That's the
That's
average.
expensive.
We have the fortune to actually work with actually natural specimens of these Greenland sharks, and what we found is that these sharks live in very challenging conditions, at high depths of the ocean, 2,500 meters deep into the ocean. At those temperatures and pressures, their proteome, for example, is extremely challenged. The way they cope with that type of extreme environment is by having a lot of urea in their body. But urea is a protein denaturing compound, element. So how do they cope with this kind of proteome instability? Actually, they recruit microbes to go back to the environment. They recruit microbes that buffer this urea-induced proteome instability through TMAO, which is a compound that actually stabilizes proteins. So we study how actually these organisms actually might have reached a super stable proteome by recruitment of metabolites that are provided to them by microbes. So the environment is definitely something you can play with to actually can challenge your proteome stability, for instance, but you can also harness sort of like tools from the environment to actually stabilize the protein.
That's fascinating. Wow. Yes, please.
On the theme of the environment, just at a human level, we've been finding major differences across human populations as well for things that, we study in the basic biology of aging. We think those are kind of universal aging processes, but across human populations we see differences. So, for example, mentioned earlier inflammaging. We've detected it very clearly in an Italian population. We applied the same model in Singapore. It's not exactly the same, but not so different. But when we go into two indigenous populations in Malaysia and Bolivia, there's no evidence of this inflammaging process. So it seems like these populations don't have anything which we could call inflammaging, right? And they don't get any of the age-related chronic diseases that we think about. They still die from infectious causes, right? But we think that it's probably something in their infectious environment, again, coming back to microbes, maybe not co-evolved microbes in the same sense, but that the environment is having major influences on which aging processes are even present.
That's fascinating. So could you speculate on why they don't have inflammation?
So, I mean, I think I want to be a little bit more precise in saying that that doesn't mean there aren't some of the genetic mechanisms like NF-kappa that are triggered in certain cells. But when we look at the signatures in terms of the cytokine profiles that we would see in any industrialized population, IL-6, CRP, TNF-alpha, these things don't correlate with each other. So in the Chimani in Bolivia, for example, they have very high levels of all of the cytokines, but they don't correlate with each other in the same ways, and that doesn't lead to chronic disease. We think that's maybe because, it's just speculation now, but they all have helminth infections. Helminths are a parasite that our ancestors would all have had, but most of us don't have. When you have helminths, you trigger a different part of the immune system, the Th2 response. This reorganizes a lot of things and is going to kind of reshape how all of that regulatory machinery is going to work. And so we think that the absence of that is maybe causing a lot of what we see as inflammaging in industrialized populations, but it's not necessarily the natural human state.
Fascinating. Wow. So there were some classic studies way back when in toxicology where you had migration of people to different countries, and do you adopt, so this was in cancer specifically, but do you adopt the local, or do you think this is genetic? So is this environment or genetic?
I would guess that this is environment, because we see the same thing in these two indigenous populations. We've got an indigenous population in Malaysia, and then there's Singapore, which is right next door. Genetically not identical, but still, the two indigenous populations are similar to each other. Also, to add to that, the Chimani in Bolivia, for them, the ApoE4 gene, which is supposed to be the biggest risk for Alzheimer's, actually protects against cognitive decline in individuals with high infection loads, right? So we see this very complex gene-environment interaction that's going on there, suggesting that our kind of simple interpretation, this gene good, this gene bad, is not enough.
Wow, that's great. Dr. Darna, do you have some thoughts here, please?
Sure. I think I really liked a new buzzword that I heard yesterday. That is the concept of exposome.
Yes.
So after the genome and the metabolomic, like the exposome. So what an individual is exposed to. And I think one of the most interesting example of that is the postal code effect, where you see the people in different postal code. They don't really have a genetic difference one to the other, but they have a difference in longevity because they are exposed to different diet, they are exposed to different quality of water, etc., etc. So I think the environment it's different. It's difficult from a scientific point of view to factor because we are used to have individuals study center on the individual and know what happens around the individual.
Yep.
But the ecosystem really plays a very important role in all of this. I think really the Western approach of looking at me, myself, and I as as the center of longevity is also will need some disruption in the future because it's impossible to think about approaching longevity by approaching the single individual and not the community of individuals.
Yeah, that's an excellent point. So we've deconstructed aging. We've talked about environment, genetics, theories of aging, trying to define aging. So we'd like to offer the opportunity to anybody who wants to ask a question. You're more than welcome to. They have microphones. So we'd be pleased to just, yep. Thank you.
That was incredible. I have questions for all of you, so I will hold back. I got one question this way. You mentioned a lack of biodiversity as aging in the microbiome. Correlation or causation?
Great question.
Or do we even know yet?
Yeah. Right. So I can tell you that, so there are studies showing also in extreme age in humans, not accounting for centenarians though, that might be like a special sort of like cohort because they are hyperprotected. There is a little bit of sampling bias to look into the centenarians, but there is actually loss of microbial diversity with age. So our hypothesis is that actually there is an active building of a complex microbiome that hosts actively recruit a diverse microbiome. to their own kind of benefit, right? So there is an active process of recruitment. There are molecular mechanisms that favor that. And in particular, the immune system might play a very important role in maintaining a diverse microbial community. But diverse microbial community might be antagonistically propriotropic in the sense that by fostering diversity, you also foster the presence of potential pathogens or pathobionts that on the long run might actually bite you back. So what we observe happening with aging is that with a declining immune system, there actually are microbes that take over the scene. So they take over the community in an ecological manner. However, so this is like microbes in this scenario are sort of reflecting immunosenescence of the host, if you wish, right? With consequences on homeostasis. However, in our own work, we transplant microbes from young individuals to old individuals. We do heterochronic microbiome transplantation to test exactly your question, causal versus kind of correlation. And what we see is benefits. So when we do heterochronic microbiome transplantation from young to old individuals, as opposed to old microbiome to old individuals, we see actually massive benefits, even to lifespan, when we do young to old microbiome. What we think is happening there is that young microbiome, diverse microbiome, is actually talking to the immune system, and so the benefits are still, once again, through the immune system. This is work in progress. This is a very kind of like new field of investigation, and this is very much what we are trying to answer. Thanks for your question. Thank you.
Great. There's a question then back there.
Thank you for the interesting discussion. I have a question, not surprisingly, about the genetic component. So if you're saying that with increasing age, you see actually that some of these genes get hypertrophy or they get increased function, do you expect that we would be able to find protective variants that actually give a gain of function of proteins and genes, or do you actually expect that most of the things that we would find would lead to a knockdown or decreased expression? What's your opinion on that? What do you think, based on evolution as well?
Dr. Cohen, I think you'd be well poised to answer.
Yeah, I mean, I guess what I think is that the impact of a specific genetic change is maybe sometimes going to be hard to predict because the rest of the system is going to try to compensate. The system kind of tends toward, think of it like in a tractor basin. So as you modify this, you still might have the same point that the system wants to get to, but the way that it gets there can be different. And coming back to the sex differences question, I think that this isn't like there are major differences between the sexes, but often the system will still tend to converge towards the same place through different pathways in men and women, right? So, you might or might not see those differences depending on which level you're looking at. But in some cases, these genetic changes will either shift where the basin of attraction is or shift which basin of attraction you're in. So then the linear prediction of what's going to happen becomes very challenging.
Dr. Muramoto.
There's a good example of what you're just posing. That is Huntington's disease is due to an expansion of a CAG or polyglutamine. And it is very clear now when you look at the human population that a repeat of 45 glutamines is the common basis of aggregation and loss of function. Yet, when individuals were sequenced around Lake Maracaibo in Venezuela, they discovered that some of the individuals, even though they had 45 CAG repeats, did not get disease until they were 60 or 70, whereas others got it when they were 20 or 25. The difficulty is pinpointing which of the other genetic variations, which are clearly familial, are protective and which enhance. But clearly, I think in the human population, this exists for diseases that have been around for tens of thousands of years.
Yep.
Good morning. I have a follow-up question on the genetics topic. So I've studied genetics since 2017. Recently encountered this concept of CHIP, which is clonal hematopoiesis of indeterminate potential. And in the recent publications of 2024 or 23, I've heard that there's an idea of opening even clinics to do this cheap therapy. And what do you think about it, about the potential of approaching all the sort of continuous mutations of the cells and sort of addressing certain genes and then saying, okay, it can be eliminated, it can be treated in one approach, basically all the diseases or like a subset of diseases.
So can you define CHIP as you're using it? You're not talking about clonal hematopoiesis, or are you?
Yeah.
Yes.
You are, okay. Okay. Anybody want to tackle that? Well, actually, I can give you a little bit of preview. I'm not an expert in mutagenesis in the hematopoietic system, but I think that's a clear example too of what we were talking about. That some people are going to carry these clonal mutations and yet have disease or not have disease, and that's fascinating and speaks to how we always get back to homeostasis if you possibly can. I do have one little insight I can offer you too, is that there's a strong tie to the environment. So using a mouse system, working with Peggy Goodell, an expert in aging from Baylor, we found that your exposure to environmental microbes actually influences clonal hematopoiesis quite dramatically as well. Not just the genetic component, but the environment. So it's going to be very, very complicated. And I don't know how to turn that into a therapy at this point. That's going to be tricky. Yeah, that's tricky. Anybody else have expertise in that? No? Okay, thank you very much. So the theme of this summit is really getting back to how do we intervene, or how do we slow aging, or how do we prevent aging. So maybe the panel, I'll ask you a few more questions to kind of bring it back to that, and then if there's more time, we'll go back to some questions. So one that's tricky as defining aging is, what is health span? So could each of you try and define it and how you would think about measuring it? What's your favorite biomarker if you were to try an intervention study starting today? So,
Yeah.
Dr. Cohen?
Yeah, so I mean, I think that we need to be thinking about health at both the biological level and then at the level of what it means for the individual in the context of their lives. So, you know, the context of your life is very individualized. We can't have one measure that works for everyone, but we need to be thinking about how we integrate across all those scales. We're in the process of developing metrics of resilience that would get at the more biological side of things, but we really want to integrate that with ideas like intrinsic capacity and so forth that would scale it across these different functional levels as well. And I think that we need to be very cautious to not just define it as the absence of disease, but quality of life and
Yeah.
With Luigi Ferrucci, who's the director of the intramural programs at the National Institutes of Aging, where he has something called the Gestalt Project, where individuals in Baltimore area in the United States go in for 2 to 3 day tests, 40 different tests to actually define you as healthy. And if you're healthy between the age of 22 and 90, you then get the opportunity to offer different tissues, such as dermal fibroblasts, or skeletal muscle tissue, or mononuclear cells that get fractionated. And together with Luigi, we're actually quantifying all of the components that define protein quality control. So our lens is really, what does it take to keep your proteome functional? And if it changes, which we know it does, when, how much heterogeneity is there when it does change, and what's the consequence? That, of course, will give us the window for rejuvenation small molecules, to rejuvenate different components, all of which we would propose are going to push back against multimorbidity.
That sounds like a really exciting work,
It's fun.
but complex. So Dr. Valenzano.
Yeah, so I very much agree also that healthspan doesn't necessarily only has to do with absence of disease but if I think about my fish that I work on, I think about how can I measure health in an individual and the window in which like a given fish is healthy. I want to think about some sort of like response, to challenges. So I want to challenge my organism as a young individual. I want to challenge the same organism as an old individual, and I want to measure how long it takes for that individual in that age to bounce back to where it was, a little bit going back to the concept of resilience. So I think Healthspan has to do with capacity to bounce back to where you were before. Even if you have, you might have some chronic disease, but if you're able to go back to where you were after a challenge, you might be in a healthier state than otherwise. If you still have that disease, but you were not able to go back to where you were. So I think we have to integrate in the concept of Healthspan, perhaps kind of like this reaction norm to a challenge.
I couldn't agree with you more, but it raises an important question. So if you're doing a clinical trial, you want to have a measure of success, and not, particularly if you were thinking about Healthspan, not 40 years later or 20 years later. So a lot of what we're talking about, or the expertise, the areas that we're studying, require functional assays or very large omics or something like that. So do you envision any way to reduce to practice some measure of health span in the near future? Because it requires often live cells or what. So Dr. Valenzano, let's start with you. It's a tricky question.
Yeah, so I can just speculate about having, indeed, some sort of ex vivo proxy for your health reaction, or some sort of deriving some like organoids that might somehow reflect your state, for example, and then challenge those in different states. Perhaps this would be very costly, or maybe accessing more integrated, higher functions that sort of reflect your thinking about cognition, for example, right? That might actually be a good proxy for stability all the way down. Can't come up with something better than this at the moment.
Yeah.
I think these two are two potential avenues.
Right. And Dr. Morimoto, from the viewpoint of proteostasis,
We start with 3,000 human genes that control the folding stability of your proteome and about a dozen cell stress responses. So from experimental data, we are then compressing and using computational to identify the best biomarker that's predictive of health and then fails in disease. It's complicated, but it's fun because it's,
Yep. Well, and we are at this stage of technology relying very heavily on single-cell seq and single-nuke seq, and that's not going to tell the whole story when you get down to cell functions. So we've got a ways to go, I think. Yeah. Dr. Adorno.
Yeah, if I can provide my point of view as an entrepreneur, that is like, okay, this is an important problem because you are not going to develop a new therapy if you don't have a way to quantify the new therapy. So if you are not able to run a clinical trial because you don't have endpoints, there's no treatment and there's no product to sell. So this is very crucial. And in the aging space is the major challenge that we are facing. And there's not a regulatory framework to solve this in human yet. But I'm very excited about the fact that last year, in a way, there has been a big step forward because a regulatory framework has been developed around aging for dogs by a company called Loyal that actually developed a new treatment for counteracting aging in dogs. And in that case, they had to face this problem, so they created questionnaires about multifunctional assessment of dogs, and testing that they were doing to actually see functionality at the end, more than a biomarker, like functionality. So I think that it's a very interesting example of how to move forward by trying to quantify some metrics in a functionality way. That's at the end is the most important. Going back to the fact of, for example, epigenetic clock that have been object of research for many years. Not always when you see an improvement in a epigenetic clock, it means that there's a net improvement in actually functionality. So we are not there yet.
Yep, that's an excellent point. Dr. Cohen?
Yeah, I think two things. The first one I'd like to say is we need to be really, really careful about the measure we develop, because as mentioned, as we talked about antagonistic pleiotropy, we might get, like, let's say, a reduction in aging when you're young, but that could come at some other cost, like increased cancer or decreased immune function 20 years later, right? So if we're going to start young people on something and we're basing that on only this one measure that we have, we need to be really certain that it's covering the full spectrum of what we want to see down the road, and that's a challenge, right? With that challenge in mind, though, we're working on exactly this problem now. John Beard, who's here, and I are collaborating. John is an expert in intrinsic capacity, so there's questionnaires, functional tests that you can use to look at things like cognition, physical functioning, and then I'm working a lot on the kind of the biological underpinnings of that, like how would you develop a metric of intrinsic health or things like resilience, which are components of that. And the idea is that even though that battery of tests that you would need with stress responses to lots of things and lots of omics is very hard and complicated, once you've measured that in a small set of individuals, you can now train an epigenetic clock or something like that to predict this future response. And that only requires one blood sample and maybe a few $100s or even less to measure it once you've got it trained. So the challenge that we want to make sure we get right, though, is to make sure that that thing that we train is a full enough picture of the health so that we don't have those trade-offs.
Right.
Yeah.
That sounds like a very promising approach, though. That's really exciting. So again, we'd like to open it up to the audience. If there's any questions for the panel, anything you'd like to ask? Well, maybe we could wrap up then by asking each of you, what do you think the end result would be if we could prevent aging or slow aging even partially? Any thoughts of what that would look like or the impact to an individual or even society, if you want to take it that far? Dr. Adorno, we'll start you.
Breaking the pattern.
Yes.
It's really difficult to imagine what is going to happen in actually having a net improvement in that. In my fantasy, in my sci-fi movie about the future, the cost associated with treatments would go down significantly. And this with free resources that can be invested in other aspects of really human evolution from other point of view.
Yeah.
Art, literature, and if we can imagine the burden of aging and diseases, it's released from us, this let us focus on other aspects of being humans.
That's an excellent point. I mean, you think about the cost of healthcare, we're not even going to be able to help the climate right now if we have to pay for both.
Exactly.
So, excellent point. Dr. Morimoto.
When we have a time where healthy aging is the norm, that will change dramatically social structures. The concept of retirement changes remarkably. Humans will have a much longer period where they can feel productive and contribute actively to society versus feeling that they only have 5 or 10 more years. So I think let's not even talk about living longer, but I like the idea of health span very much.
Yep.
That instead of, from 55 to 70 being a period of looking around and saying, oh dear.
Yep. Absolutely true. Dr. Valenza.
Yeah, so I think I was reflecting on this, and I was thinking that I think we have a glimpse already now for those who are probably in the late 40s and over in 50s. Probably we already have an experience of a society where people over 60 and 70 and 80 have more agency than they used to, 20, 30, even 15 years ago, to be honest. So I think that already now we sort of witness to some extent sort of more participation, more active participation of people in the older age. So perhaps that's something also to be learned in what has happened in the past few years and what could be even a demographic explosion of that portion of the population. I'm not sure it's necessarily great for society in the sense that, you know, shift towards like a more agent, late life portion of the population might lead to more conservative kind of like shifts that
Politics,
might
too.
lead to dramatic shifts. But of course, the hope is that also selfishly to stay, you know, to have the capacity to contribute positively more to society. I think it's a very difficult question.
Yep.
I'm
Yep.
not sure.
Thank you. Dr. Cohen.
Yeah, so I guess, you know, the world I would like to envision is one where maximal lifespan, human lifespan hasn't changed much, but we're really healthy right up into the end, and then things go downhill very quickly at the end of that. And I do think that we can get somewhat close to that. I think again of the Chimane in Bolivia, and they have infectious diseases that we don't want, but they don't have the chronic diseases that we fear. Can we figure out a way to kind of have our cake and eat it too with those things? Maybe we can, right? So like at a health level, I'd like that. But I think, you know, we're mostly into the biology here, but I think we need to not forget the social aspects of it. We live in societies where the value of grandma has gone down as a repository of knowledge. We've got Wikipedia instead. And so the value of being an older adult is less than it was socially. And there's now a stigma of being old. We just want to try to replicate youth, which if you think about, you know, many other societies, ancient China, for example, being old was venerated, right? And so this changes your perception of yourself as you age. And I would like a world where older adults feel that they're valued, feel that they're contributing, and I think that is actually one of the best interventions for their health.
Yeah.
But that requires a major societal change and a shift in how we think about the aging process. We don't just put people out to pasture at age 65.
Yeah. Well, these are all excellent points, and I guess the take-home message is we need a lot of research in basic aging biology so that we can have a way to intervene in order to really change society and the world for the better. So thank you all to my esteemed panelists. You've been great, so thank you very
Thank you,
much.
Laura.
Thank you.
And thank you guys for coming. Really appreciate it.
Thank you.
Thanks.
At the end of my presentation, I will specifically go into gut-brain axis. So we really, in my group, work at the interface between microbiome, host interactions, in the context of aging research specifically. So let me introduce first the way that we are thinking about the aging process in my team, so which is, I hope, shared with many of you. So, aging is this kind of like phenomenon that affects multiple levels of biological complexity, from molecules, and it has really to do with damage and damage control, damage management, if you wish, right? So damage can happen at multiple levels of biological complexity, all the way from molecules to, large complexes that deal with, this is a proteasome system, so that deals with protein misfolding and damage and degradation and recycling. Damage can happen at the level of tissues, and then the organisms can deal with this type of damage in different ways. There are like specific immune responses and that lead to damage control, and damage can happen, insult can happen from parasites, pathogens, but damage can also come from intrinsic processes, cellular processes, intrinsic, so not only extrinsic processes, but also intrinsic processes. Now, how organisms deal with this continuous damage coming from outside and from inside is also oftentimes what determines how organisms age, how long they live, how broad and how long their health span is. So what we are interested specifically in my team is understanding how different model organisms or different species in the world deal with damage, and maybe by harnessing solutions to damage in different organisms, we can develop new therapeutics. So a very useful framework to think about all the things that might be changing, and that can be also addressed for improvement during the aging process, is actually the very famous hallmarks of aging that's been recently upgraded, updated, adding some important elements to this discussion, including dysbiosis. So what I want to lead you to now is to think about the way that we have been classically thinking about aging and all the things that might be going wrong during aging, but also that can be targeted to improve the aging process, might actually be beneficially impacted by the interface with the microbiome. So let me guide you through this. So as we think about, like progression throughout different life stages, we know that actually the environment can play a very important role into modulating the pace of aging. And so we know that diet plays a very important role. We know that actually drugs, as an environmental intervention, if you wish, as an external intervention, can impact the aging process. Exercise is a very important intervention that can affect the aging process. And also temperatures. There are different studies that see how temperature levels in not just in cold-blooded organisms, but also in warm-blooded organisms can buffer the aging process. Now, what I want you to kind of like lead you to is in this slide, is that a very important interface between the host and the environment is represented by our microbiome. So this is a very complex community of microbes of different types that actually help us interface ourselves with the environment.
microbes are like this permeable membrane that we are surrounded by, and that translates signals from the outside in, but also from us to the environment. So microbiome actually contributes virtually to all biological processes that we know of. And I will just list a few here. So from nutrient absorption and processing to immune system maturation to even some organisms like squids, even inter-individual communication. There are these fluorescent bacteria, bioluminescent bacteria that actually help communicating. So microbes really have been associated to many processes. And our point here is that since they contribute to virtually all biological processes we know of, they might also play a very important role during the aging process. So there are several groups at the moment, actually a few groups focusing specifically on host microbiome interactions in the context of metabolism specifically. And I would like to highlight particularly two of my colleagues, Lisa Meyer in Tübingen and Felipe Cabrero in Cologne. They are specialists on gut microbiome, Felipe particularly in the context of aging. And so what they really study is, for example, in the context of drug, the way that we deal with actually drug uptake, microbes actually play a very important role in actually drug metabolism. The way that actually drugs can be modulated, can be absorbed, actually also bioaccumulated in cells, it's strictly dependent on our microbiome. And also microbes respond to drugs by metabolite secretion, and actually responders, non-responders to specific drugs might very well actually, and do actually account on different microbial compositions. For example, in the case of metformin, it's been shown by my colleague Felipe Cabrera in Cologne that actually, like in C. elegans, for instance, that the impact of metformin on longevity and aging in C. elegans is massively mediated by microbial intermediates. So what I want to highlight now is that one of the most kind of like charismatic, if you wish, one of the most convincing anti-aging drugs we know of, rapamycin, is actually a microbial metabolite. So let's not forget about this. So this is just to highlight the fact that actually the microbiome is not only like something that interferes and intervenes and mediates drug impact on health, but also actually microbiome could also be like a source of metabolites and compounds that might help us in combating the aging phenomenon. So I want to tell a couple of words about our institute. So we are located in Jena, in Germany. This is a picture of Jena. It's a beautiful city in the middle of Germany. You're welcome to come visit anytime. We have at the Leibniz Institute on Aging a specific focus on microbiome and aging. Actually, we have like a research focus on microbiome and aging, and actually we have 3 wonderful junior PIs that are focusing on different aspects of microbiome and aging, Melika Donner-Trash, Clara Correa-Melo, and Kaja Vinek, covering different aspects of microbiome and aging. So we actually also organize microbiome and aging international conferences every year. So we have one also later this year in 2025 in the fall. And actually Hevolution has contributed also to this conference. So really thankful to the contribution of Hevolution to this conference. So our interest in microbiome and aging actually has to do with, is multifold. We're interested in how microbiome changes throughout aging of the host actually can be used to understand how the host is aging. So by drawing from the microbiome features of aging of the host, we actually look at microbiome as an entry port to interfere with the aging process. So like as a polypharmacy to interfere with the aging process. So this twofold difference. And so in other words, going back to the hallmark of aging, we're very much interested in how microbiome can actually impact all of these hallmarks. So if you think about actually chronic inflammation, for example, stem cell exhaustion, cellular senescence, mitochondrial dysfunction, et cetera, one of our specific focus is to understand how each of these hallmarks actually is or can be modulated through the microbiome. Is modulated in the normal aging process through the microbiome, but actually also can be engineered to be modulated through microbial mediators. So our specific research model that we mostly focus on is this colorful fish. So we work on comparative biology of aging, and killifish our premier model. This is a short-lived organism that lives just a few months, has a lifespan of about 4 to 6 months, depending on the strain. And what's really peculiar about this fish is that it undergoes in a very short amount of time several age-dependent transformations that really resemble how we humans age or other mammalians age. So this, of course, is not a mammal, it's a fish. I will list just a few of those age transformations in the killifish, and then I will go back to the microbiome. So killifish undergo actually bone density loss with age. They undergo also sarcopenia. They have actually neoplastic transformations like melanoma present in this tail here. They have fibrotic, scars increasing with age spontaneously. They have fibrosis, which is a hallmark of inflammation. They have cellular senescence, spontaneously accumulating in just a few months. These are actually coronal sections of the brain of the killifish. This is a young killifish, this is an old killifish. The blue staining is actually cellular senescence accumulation. Killifish undergo increased protein aggregation spontaneously in the brain. Killifish also undergo immunosenescence. So B cells in killifish, so just like us, they have B and T cells, undergo actually antibody diversity loss spontaneously as a function of age. And this plot shows actually the antibody diversity of young killifish and of a cohort of old killifish. So there is a dramatic decrease in the naive, in the pool of naive B cells, in circulating cells. So a postdoc in the lab in the past studied actually how the microbiome, whether the microbiome is actually dramatically changing as a function of age. And what he discovered is that actually as killifish get older, the microbiome richness, the microbiome species diversity drops dramatically as a function of age. So this actually has nothing to do with young or old fish having more or less microbes. What actually is really changing is the number of microbial species present in young versus old fish. So old fish have fewer microbial species. And so we ask whether this is simply like a correlation of the aging process, or this actually has an impact on the aging process. So what Pat devised to do was to do actually heterochronic microbiome transplantation, transplanting microbes from young to old killifish. So what Pat found is that when we transplant microbes after antibiotic treatment from young killifish to mid-age killifish, and we compare it to killifish that receive microbiome after antibiotic treatment from the same group, and then we have another control of only antibiotic-treated fish, what Pat observed is actually we have extensive extension of lifespan in killifish that acutely, just once in their midlife, undergo a heterochronic microbiome transplantation. So this is a dramatic lifespan extension. Not only there is a lifespan extension, but it's also behavioral improvement in their locomotor activity, and as I will show you in a couple of slides, also in their brain function. So in learning, in cognition. So we did extensive transcriptomics of the killifish gut, and what we observe is that a lot of anti-inflammatory terms are actually systemically up in killifish receiving microbes from young donors, as opposed to same age donors. Of course, the big question is what are the mediators of this effect? But the good news is that actually colleagues from Cork in Ireland studying mice actually found very similar results to what we find in killifish. And so the research in this field is really exploding right now. So the mediators is the big question, and long story short, we don't have an answer yet. So we don't know what mediates the effect of heterochronic microbiome transplantation into benefits to host physiology and aging. So this is something that it's like a really new field of investigation, and we are really at the beginning of understanding what's going on. Just to conclude my presentation, a postdoc in my team, Dr. Dennis De Backer, is actually studying the impact of heterochronic microbiome transplantation in the brain function, again in killifish. Why we use killifish again? Because it's in a short amount of time, which is experimentally accessible, recapitulates several aspects of the aging process in vertebrates. So this is again a coronal section of the killifish brain that shows different cell types. These are neurons, magenta, these are nuclei, and this is actually astrocytes. And so focusing on this area of the brain, what Dennis actually saw is that actually there is increased microglia activation with L-plastin in killifish brains spontaneously with age. And while actually in normal killifish age old, there is actually increased gliosis with astrocytes increasing in surface, when you do a microbiota transfer from young donor, actually gliosis actually declines dramatically. Also L-plastin, so this kind of like microglia activation declines, but there is no change in actually neuronal loss compared to the controls. So what I want to finish with is actually, again, preliminary work on killifish behavior. You can train killifish and you can study learning and memory in a species like killifish. You basically condition a fish to learn to associate a light onset with an adverse stimulus, bubbles, that they would hate. And so they learn that every time they're cued with light, they would go to the compartment where there is no light, where there are no bubbles. So they learn to go to the safe dark compartment. So if you expose fish to this type of associations, you can actually see that actually the performance, so the number of time that fish avoid the light after this association actually looks like this along cycles in young fish. And then if you see aged fish, this is the performance. So you have a better performance in young than in aged fish. This is all you need to know from now. So what happens if we do microbiome transplantation? And then we ask in old fish, how do they learn this task after a microbiome transplantation? Well, actually it turns out that actually old fish that have received young microbiome transplantation have a performance that starts off like the age-matched controls. So they start pretty bad in their performance, but actually they jump up and actually they are able to learn and reach a performance which is very comparable to that of the younger counterparts. So heterochronic microbiome transplantation actually is boosting learning performance, at least in the early stages of this trial. Then there is a drop-off. We don't understand whether this is fatigue or is this really, we don't think this is memory loss. But this is just lasts like 50 minutes. Interestingly, also fish that receive gut microbiome from age-matched controls also have a slight improvement in learning as opposed to the untreated controls. Now, if you treat fish with antibiotics, the learning performance actually stays in the middle. They then don't learn as well as the ones that receive a young microbiome transplantations. Okay, so this is the type of research that we are doing in our team. And just to conclude our framework is that actually microbiome actually does have an impact on hosts dramatically. We are studying the mediators. We're also studying actually how the immune system actually is shaping actively the microbiome to build up a young, rich microbiome in a young individual, healthy individual. However, we also understand that aging in the immune compartment might have a dramatic impact on the microbial community. So we are working now to sort of like targeting immune system to rejuvenate the microbiome itself with hematopoietic stem cell transplantation, for example. Just to conclude, last slide of data, we're also very much interested in understanding how different species, short-lived species and long-lived species, including humans in the middle, how their microbiome may be actually mediating to some extent some of the effects that lead them to live such a long lifespan. In particular, for the greenest shark, we have a very good first hunch into how the microbiome might be playing a very, very important role in sustaining life for a long time. With that, I would like to maybe tell you that killifish community is exploding, expanding in the aging sphere. We have a symposium later this year. And this is just thanks for my group, and this is to the funders. And with that, I would like to thank you for your attention.
Sorry for some technical issues. Thank you, Dario. It was fantastic talk. And I've never worked with fish myself, but it's so cool. And just a question, I think I talked to you. Have you ever, yeah, that's the one, yeah. Have you ever looked at the microbiome on the skin microbiome?
I don't know if my mic, yeah, so not actually, no. We specifically looked at intestinal microbiome so far, but I think it's a brilliant brilliant idea. We should definitely look into the skin microbiome, how that differs from the
Because
gut
they have
microbiome.
really strong microbiome because they live in the water. Yeah, it's cool. Thank you for being here, and I just wanted to give a little insight on gut-brain axis. His talk made my talk very easy. I kind of put together some work that we do in the lab, and I was told that students will be here, so I kind of prepared it for the students so they can take home something. Gut matters. Everyone says, and especially when you talk about gut-brain, people who work in the neuro side, they always thought that the issues that happen in the brain is above neck. But what is going on in the system, like below in the peripheral organs, is so important, which kind of maintains your overall health. And I see bacteria especially as our forefathers. Like when you look at the evolution, bacteria came much ahead of us, and then we evolved slowly. And people are talking about aging here. What is aging? So a baby is born sterile, completely sterile, and then they go through this phase of constantly educating and fighting with the environment to live. And when somebody dies, the first thing that happens is microbiome within your system, where the intestine is fully colonized with all these organisms, they start eating the body from inside. So we kind of decompose. I don't want to put that real fact forward, but that's what happens in reality, and how our body kind of educates itself over time to maintain its bridge with the microbiome. It provides something for the microbiome to exist, and microbiome provides something for us to kind of have the symbiotic relationship. So I just wanted to show some stuff that we work in the lab. Yeah, this is one of my favorite slides. When a baby is born, like we are like a sterile slate, like empty slate. You can write whatever you want, and from the mother, the microbiome gets shifted to the offspring, and then you take up on yourself how you live your life and how you're going to provide an environment for this microbiome to colonize. So recently, we are also looking into advanced maternal age. Apparently, the mother's health is playing a very key role or very important role in educating kids' immune response through the microbiome and its metabolites. We very much focus on umbilical cord blood, what kind of metabolites are being transferred from the mother to the child before the child is being born. And once the child is being born, it kind of colonizes with very specific bacteria. There are zillions of bacteria going through the system, but the system only allows specific bacteria to colonize the gut because of the dietary presence of breast milk. It's usually, I don't know whether people have come across bifidobacterium and lactobacillus, which is called probiotics, but that is being isolated from the mother's breast milk or the baby that drinks only the breast milk. So eventually, 3 months after a child is being born, the microbiome changes. 1 year after, the microbiome changes, and then it goes through an evolution within you, just like how you grow. And when it is very fascinating to see, and we use mouse models. When you take a mouse that lives in the environment, in a regular environment, they actually live only between 17 to 20 months. And when you take the same animal, house them in a laboratory under pathogen-free environment, in the sense there are bacteria, but there are no pathogens being exposed, they can live up to 30 months. But when you take the same mouse and house it under germ-free situation where there are no bacteria, they die at 20 months. So microbiome is important because it's constantly educating our system, but what kind of microbes we are being exposed to, I think makes a key role in how we age and how our immune system gets matured. Little bit on bacteria, I'm very fascinated with it. There are two forms, gram positives and gram negatives, called firmicutes and Bacteroidetes. These are the major players, and on the other side there is minor phylum, which is actinobacteria, proteobacteria, verrucomicrobia, and there are so many that are constantly being discovered. And it is very important to know that bacterial cell numbers actually are more in your body than your own cells. So they are existing there for a reason. And that reason we see is constantly educating your immune system. Especially in the COVID world, everyone were using masks. And for 2 years, we were not exposed to our regular environmental pathogens. When people removed the mask and started living, even the regular virus which infects us is very potent, and we cannot overcome that. So because there is a snapshot in our lifespan that we are not being exposed enough to that environmental microbe. So there is a very famous book written by a Canadian professor. He says, let kids eat dirt or play with dirt. I think it's very important. We live a very sterile environment now. We call civilized, urbanized environment, but I think we also need to expose ourselves into some of these microbes, not pathogenic environment, but more a general natural environment. I just don't want to explain it much. Dr. Dario did a great job, but I just wanted to point out that gut microbiota is very, very important. Oops. Very important, especially because it is a key player in educating your immune system, and gut is the largest organ that holds different kinds of immune cells. So there is a reason why it exists, and obviously nutrition. So microbiome diversity is only through your nutrition. What do you eat? Do you eat high fiber food? The fibers are very important for bacteria to cling, because it's like an open tube. You put something new in, the old stuff gets washed out. How can you retain the good bacteria in? By providing some kind of products that they can eat on and they can grow. So that is your fibers. So eat good quality fibers, but that doesn't mean your food should all be high fiber. So they provide the environment for these bacteria to colonize and secrete all these beneficial metabolites that can directly communicate with specific immune cells and activate them in a very specific pattern. And we look into those patterns. This is another slide, how gut is important for brain activation. I don't know how many people, when they are stressed, their gut seems to not work fine. They need sugar. So obviously when you eat chocolate, for example, you feel happy, you feel like rewarded. And a lot of people do behavioral studies where they treat these animals by giving food, and they feel rewarded and they do what they want them to do. And it's very important to kind of keep your gut healthy. And at the same time, I don't know whether people come across this name called happy hormone, which is serotonin. Serotonin is produced by a specialized cell in your gut called enterochromaffin cells, and they secrete serotonin into the gut through the diet. They get reabsorbed, and through that cycle, it kind of maintains serotonin levels. So many people experience depression, and they follow that depression with eating more or eating specific kind of food, which kind of keeps them happy. And it's a great, amazing thing that serotonin is so important in the brain, and 90% of your body's serotonin comes from your gut. And we do look into how microbes play a key role in regulating one of those pathways, and that's kind of how I got the funding last year from Hevolution. This is another slide where how the gut microbiota kind of works with the brain. Usually, all kinds of microbes are present within all of us. So people always use this term dysbiosis. Like, what is that? It's not yet a field where, oh, these are the bad bacteria, these are the good bacteria. Nobody knows yet. People do know good bacteria. People don't know yet what are the bad population, except couple of pathogens. We all have them within us, but at a certain age, when they age, the composition shifts. And when you look at a healthy person and an aged or diseased person, the composition of the microbiome has changed drastically, and we coin that term as dysbiosis. We are still dysbiosis is still an evolving field. We are yet to discover what kind of patterns exist and how can we tool that system to benefit the humanity. Inflammaging. So we have talked about aging the whole day yesterday, most of the day today. This is a term that when a person ages, if you take a blood sample from a young person and an aged person, you just look at some pro-inflammatory markers, you naturally see that aging has an increase in pro-inflammatory response, not like super high, but a chronic low-grade inflammation. We believe that inflammation is because there is a barrier being breached within the system, and that is playing a key role in elevating these inflammatory responses in the body. I'll show some of the slides how we measure them in the next coming slide. So just for people who don't know how the gut looks like, it's a very fascinating organ. It has a specialized cells called absorptive epithelial cells outside. I'll just kind of go. So these are absorptive cells, and this is where the stem cell niche is. It's very interesting organ because every 7 days, all these cells will die off and new cells are constantly replenishing. And this happens in any normal human's body once every 7 days. And in aging population, we see that this recovery is going down. It's lost. And you see these specialized cells. So there are goblet cells, there are enterochromaffin cells, which are secreting serotonin, and these are stem cells. Bacteria can actually come all the way till here, but there is a very strong release from Paneth cells and stem cells to keep the bacteria at a distance. And there are goblet cells which produce these mucus, and that mucus is constantly being produced, eaten by bacteria. By eating the mucus, bacteria induces the cells to produce more mucus. So that mucus gets built up and flushed out. And usually in a healthy person's intestine, bacteria resides very much on the top. And consider this to be like a circular tube because this is just a coronal section of the gut. Your material is moving there, and the mucus is kind of covering. So some bacteria can eat this mucus, come in close contact with the epithelial cells and regulate immune response. And your immune cells are seeded through the lamina mucosa, and there are nerve bundles, blood vessels running throughout the system, and nerves, the vagus nerve or afferent nerves, so they run along this serosal side and gets connected to the brain. And what happens here, you eat, like gut is one system we are constantly abusing in the sense we eat, we drink, everything is being absorbed, processed, and sent out. So through that process, bacterial products are also being absorbed. How much of these products are being absorbed is varying between young population and aged population. And I think that is playing a key role in how your immune system is evolving when you age. And that is just a staining from the mouse, how the goblet cells are like oozing out mucus, and it kind of forms like a nice jelly layer around it to protect the microbiome invasion. So this is another slide where I just want you to take a moment and understand that this is nucleus staining. So I just want to go back. So consider this as the blue color. So these are the blue cells, and this red is the biomass, which is the microbiome that is blooming in your gut, depending on what you eat. And if you look at it, these are young animals, these are aged animals. So these are 3 months old, these are 20 months old. So in the human terms, it's equal to about 65 years old animal and humans. So you can nicely appreciate that bacteria is doing something to us, but they have a very clear distance from where our host cells are. And with time, you see that that barrier gets completely breached, and the bacteria slowly starting to come much closer and sits right on top of our cells and interact more. In this situation, the bacteria directly doesn't touch the cells. What they secrete touches the cells. Under this situation, the bacteria directly touches the cells, which is a direct activator of pro-inflammatory responses. We are still trying to understand what are these specific bacteria that can pass through the barrier, because they like anaerobic environment. Our intestine is completely devoid of oxygen. But when they cross that barrier, this area is oxygenated. So mostly pathogens can shift between no oxygen situation and yes oxygen situation. So we are understanding how they are eating up the mucus and coming in close contact with the host cells, and what kind of products do they secrete, and are they being visible in the blood? So those are some of the research that we are working on. And this is just protein staining. Again, I wanted to highlight that intestinal epithelial villi, and you see that brown staining. They're called E-cadherin. It's a tight junction protein. It's like door to your house. If the door is closed and you have the control who to come in and who to go out, then the system is protected. If the door is not working fine, anybody can walk in, anybody can go out, then that depends on the environment. If the environment is good, then you're protected. If the environment is bad, then you're not, like anything can happen to you. So this is a young situation, and that's an aged situation. Obviously, in aged mice, we see expression, but the expression is so much significantly reduced compared to a young animal where those doors doesn't work properly. This is just to test or to show or highlight how strong the gut and brain communicate with each other. We had a model, we caused injury in the gut within 24 hours. These are the goblet cells I was talking about, which is stained differently, which spits the mucus. Within 24 hours, you still see the goblet cells, but they are completely lost. So when there is an injury in your body, your gut responds immediately. So we think that when people are aging, earlier injuries that they have gone through, maybe there were snapshots of moments where they lost that barrier, they were exposed to this pathogenic environment, there's a shift in their immune system that might age them faster than people who never had any kind of injuries. So this is just a model just to show that how strongly gut and brain is constantly communicating. Now I'm shifting a little bit from gut to brain because we talk about gut-brain axis. So you can very clearly see this is a young brain. It's a mouse brain looking at the cortex. And this is an aged brain, 20 months old, 3 months old. And the green colors that you see are microglia, which is the police force of the brain. They are the immune cells that kind of protects the brain from any kind of pathogens, any kind of infection. And you can very clearly see a healthy microglia will be more ramified. It will have all these tentacles, but inflamed microglia will kind of crumple up. It becomes like a ball, and it only spits pro-inflammatory signals. And in young animal, we see this nice branching out of microglia. But over time, when the animals age, I wouldn't say that all microglia becomes amoeboid, but we see a strong, significant population of microglia becomes more amoeboid, and they tend to show more pro-inflammatory signals than anti-inflammatory signals. This is just on the microbiome. Again, I told that the big players are firmicutes and bacteroidetes. Young microbiome, old microbiome, we do see that bacteroidetes gets lower, firmicutes goes up. I wanted to take a moment and say that this situation is different when you do the same same experiment in European population. They have a shift in firmicutes to bacteroidetes. So we believe that probably climate, diet, all of those factors are playing a role in how this population is decorating our immune system and how our immune system kind of responds to these niche which is in our system. This is just again showing or highlighting what kind of bacteria are present. As I mentioned, green is the young. You can see Bifidobacterium is much, much larger in number, but when they age, we lack them. Bifidobacteria in specific are really key modulators because they are most likely the very first ones which colonize the baby's intestine, and they produce some of the molecules which are mostly tryptophan based. They can pass through blood-brain barrier. We have done some germ-free experiments. When there is no bacteria, you just don't see that product in the body, and when there is bacteria, you see that this molecule can be present in the brain, and they very strongly communicate with a specific receptor that activates and suppresses. It's like a sword. It can be used both ways. The same pathway, if the same receptor gets activated through a host-derived molecule, it becomes more pro-inflammatory. If the same receptor gets activated by a microbial metabolite, it becomes anti-inflammatory. It's very unique. It's called aryl hydrocarbon receptor, which is predominantly present in microglia and other cell types too, but we are looking into how microglia responds to it and how microglia matures in the presence and absence of this metabolite. Everybody has said now we are also looking into senescence, and this is very interesting. If you take an endothelial cell which is decorating the blood-brain barrier, a young endothelial-derived conditioned media, treat them on aged cells, you can prevent or you can reduce senescence. The other way around, it increases senescence. And we very strongly think infection, pathogenic invasion has a strong impact on increasing senescence. I'll just stop here. This is like one final slide. It's like a story how I ended up in aging research. My lab primarily looks into Alzheimer's disease, and we had aged animals that we had to sacrifice because they were beyond 20 months. We don't use it for our AD research. And we had a bunch of animals which were 27 plus months old, and they were surviving 30 months. They looked all fine. So we were like, okay, let's just look at how their microbiome and the immune system looks like. And we found the ones that survived had a very strong population of lactobacillus, and their immune system looked much more anti-inflammatory than pro-inflammatory. So we decided to kind of go with the data and then delve into aging and how microbiome can shape aging. Yeah, you can see how it's like a beta diversity plot, how they are distinctly different. Young population, aged population, super aged population. So these are the ones that kind of like started dying. These are the ones that survived beyond the time that they are expected to be alive. And yeah, I will stop here. It's been great. And I wanted to also thank a couple of members who are instrumental in getting all this data, and all my collaborators from Texas, especially Dr. McCullough, who is leading in sex differences and cerebral vasculature research, and all the funding organization, and definitely Hevolution through AFAR. I got funding last year to do some of those last data that I showed for the next 3 years. Thank you all. Thank you.
Thank you.
Any questions?
I think it's over. The
It's
session
done?
is over.
Oh, okay.
Yeah.
Did I take that much time?
I spoke 30 minutes. The session is over.
Okay.
I spoke 30 minutes.
The next people are already waiting.
Declan is almost 2 years old.
I'm a pediatric biochemist. society. So a lot of the discussions here are really looking at the cellular changes that are associated with aging and the impacts they can have on our health. What we're interested in that, and some of my work looks at that. I'm particularly interested in how that impacts on people's functioning, their intrinsic capacity and their ability to be and do the things they value. And then what we're also interested in is how that impacts on society. So I think that does connect all these dots, so even though they are very diverse.
Well, I guess, they are diverse, and I have a similar existence myself when I'm covering new cellular discoveries or new investment rounds or whatever it may be. And just interested in your thoughts on coming to the Hevolution event as you have. Do you feel that this is a different type of event to everything else that you've been to?
Yeah, I've been to aging events for 20 years now, and they span that whole range through from cellular through to societal sort of impacts of aging. What this reminds me of is the World Economic Forum, actually, and I think it provides a totally. We've really leapt into a new dimension with this Hevolution investment and these sorts of events, and it allows us to really think of these things in the bigger picture, because in the past most of the research on science was just research on science. Most of the research on society was just research and meetings on society, and this enables us to have discussions that look at the bridges between those components.
Yeah, and it's quite refreshing to see senior politicians and members of the country's leadership talking about health span, longevity and so on at a policy level, which is starting to happen now, which I guess is...
And so impressive to see how committed they are and really genuinely interested. Sorry.
Yeah, so I guess let's jump in, shall we? So in terms of, we've heard the government spokespeople talking today. How can we develop a clear and really coherent health span strategy that resonates with policymakers and the public? Because there's is it the healthcare argument? Is it the finance argument? Is it the socio-economic argument? How do you feel we could shape that narrative?
Okay, I tried to do this for 10 years at WHO, and it's a long haul, and it's hard. And it's hard because people have these very embedded stereotypes on aging. And they still think of older people as a burden on society. All the economic impacts are on the costs that's going to impose as populations age, and how we can minimize those costs. People don't look at the contributions, and because they don't look at the contributions, they're not so much thinking about the investments we can make that will foster those contributions. And health, obviously, is one of those critical components which we can invest in, which will really impact on the subsequent consequences for individuals and for society.
Yeah, because I guess when you think about flipping the model, right? So we've got every country has an aging population now, which is growing. They don't have the population pyramids of some countries are pretty horrible, as we know. They don't have the tax base coming through. So how do we start to measure the impact of the fact that people perhaps won't be retiring at their national retirement age? They may not have the finances in place to be able to do so. What are the sort of metrics that you can say that actually, if you invest in health span and aging research, these are the economic benefits that you're going to see as a country, and here's the downstream tsunami that you're going to potentially avoid?
Well, first of all, I would question whether those demographics are horrible. I actually think they're good. What's horrible is that society isn't adapting to them. And in fact, we also carry just so many myths about the implications of those demographics, and they limit our ability to create a new society. I've got some slides in a little while that I can show how in fact, so far, the impact of population aging on macroeconomics has been very minimal.
Right. Even in the US, when you look at the amount of money that's spent on aging diseases, for example, nearly $4 trillion. Is that something that you would say is factored in, or is that something
I think it's 7% of increasing healthcare costs are due to aging.
Right.
The rest is due to inefficiencies.
Right.
And new technologies.
Okay.
And yet it's an easy political sort of platform to blame older people and try and cut expenditure, when in fact the problem is they have a dysfunctional system in the United States.
The UK is a prime example of a completely broken system.
Yeah, well, it's not as broken as the US.
Is it really? Okay.
Well, in terms of it costs about 40% less. with the same output and the same outcomes. So yeah, it's probably not as broken.
Oh, okay. Well, that's good to hear. So sort of getting back to the question of metrics. I mean, you're representing an industry, and obviously to policymakers and so on. What are the baselines that you actually start to count now? How do you demonstrate this through statistics and figures?
Well, one of the big problems is economists don't actually even collect the statistics.
Right.
You know, the key question here is how do we get the best balance between the contributions of older people and the costs of older people?
Right.
And they don't even collect information on the contributions.
Right.
You know, older people contribute in many, many ways, and yet we just look at the potential impacts on healthcare systems, on pension systems, and so on. But in fact, the overall balance is in the favour of older people contributing more than they take from society.
And is that because of their potentially their boomer wealth, or is that because they're actually still out there working and paying taxes and contributing to their own pensions and so on?
Older people, first of all, contribute by, many of them are still in work, so they're paid employment.
Yep.
Secondly, some of them are self-funded retirees. Older people pay tax. The older people pay more land tax and about the same so property tax,
yep.
and about the same consumption tax as younger people, and in fact they pay more tax than people under 30 years old. And we don't talk about those under 30 years old as if they're a burden on society.
No, okay.
So you know that when you actually look at the data, it's very different than the stereotypes which we hear every day.
Right. Okay. And so
and older people also contribute in other ways. Like older people can be innovative. The most successful entrepreneurs are people over 50. It's not young people. It's people over 50 because they've learnt and they don't make the same mistakes.
Yeah.
And yet, you know, this just runs counter to all the stereotypes which. You know,
well, it's very interesting. There's a new VC fund based in New York that only invests in people over 50.
Oh, I have to look at that.
It's good. And of course, you know, I mean, I'm an entrepreneur. You know, I started my current company when I was 49, right? So,
yep.
I mean, I guess that's the point where,
yep.
you know,
exactly.
not the first rodeo and so on.
You've learned along the way.
Yeah, that's right, got the scars. So in terms of the, just moving back into the, you say from cells to whatever, in relation to your analysis, what should we be looking at in terms of biomarkers and indicators to understand whether a country has got a healthy aging population, or has got an aging population that perhaps needs attention? I mean, obviously, we see the disease statistics, which is, you know, that's very easy to see that because those are, you know, data that come through hospitals. But how else can we measure whether a country's moving in the right direction or not? I mean, is biological age something that
how can we measure health span? Is what
yes,
you're asking.
Yeah.
I mean, it's interesting because we say health span, but what I hear between the lines here is we're talking about disease-free span, and that is number one problem. We need to actually be quantifying health.
Yeah.
And how do you do that? It's not straightforward. When I was the director of aging at WHO, we came up with this model of healthy aging, which looked at health from the perspective of people's ability to be and to do the things that they value. And that's actually what older people, that's what they look at when they think, well, what's important to me? It's can I still do the things I want to do?
Yes.
And so if we can quantify that, then we're starting to get much closer to health than whether they have disease or not. And at WHO, the way we framed it was that results from individual attributes, what we called intrinsic capacity, and it's a term which people are using more and more.
Yes, I'm hearing a lot of that.
Yeah. And the interaction of the individual and their environment, which gives them the ability to do what they want to do. So you need to quantify each of those things. You can look at the environment and issues like, you know, is there disabled access public transport? Is there mandatory retirement age, which these barriers which prevent people contributing? But you can also look at the individual, and I think at a biological level, intrinsic capacity is a much more logical outcome, which we should be linking all these biomarkers to, than the presence or absence of disease.
Yeah. So how do you measure intrinsic capacity? What are you looking at? Is it a combination of qualitative and quantitative data?
No, absolutely quantitative, right. Totally objective.
Okay.
And essentially performance measures. So the way we traditionally frame intrinsic capacity is express capacities such as cognitive capacity, locomotor capacity, sensory capacity, vision and hearing, psychological capacity, your psychological state, and then underlying that a domain which captures resilience and energy and those sorts of things, the underlying drivers which allow you to have those capacities and to maintain them.
And is that based on blood data, or is it purely about questionnaires and so on?
Well, no, most of it would be objective testing. So for example, you can measure gait speed, chair rise for locomotor, you can do cognitive assessments, you can obviously test vision and hearing. Psychological, you generally would do it through a questionnaire. But so most of it is objective. And one of the challenges we have is to link all the things we're talking about here, the biomarkers, the methylation, immune status and things like that, with those functional outcomes. And that will give us a totally transformative way of thinking about health.
Is that happening at the moment, or is that a job still to be done?
I think it's just starting. It's work we're doing at Columbia. There's also at the moment an ARPA-H call for research to do exactly that. And I think that's very exciting. And if we can get those endpoints, the performance endpoints, the functional endpoints, and then the surrogate biomarkers, if we can get them approved by FDA, then that gives us outcomes which can be outcomes for clinical trials, and also they then provide the opportunity for looking at how health systems are financed and how clinical practice is practiced, bringing in the practice side of all of this. And when I was at WHO, we came up with a program called Integrated Care for Older People, which is around care pathways for people, which is based on those functional outcomes as the entry point rather than disease.
Right, so that work hasn't happened yet, it's on its way. Do you feel that there's sufficient confidence between the policymaker, the clinical community, the research community, the scientific community on really measuring those baseline markers? I mean, obviously there's the Biomarkers of Aging Consortium, which is kind of getting to the point where it's got nearly standardization of these. It's not there yet. So do you feel that the time is we're able to do that now, or is that still...
No. And one of the frustrating things is, if we look at most of the research in this field, it's segmented into people working in each of their little domains.
Right, yeah.
And bringing it together in a more holistic way, which is the fundamental principle of geroscience, is sort of lacking. So I think we need some way of putting in place that cohesion, and I think these functional measures are the way to approach it.
And is that your organisation's role in doing that, or do you feel
it's not our role? We do it, but yes.
Right, got it, great. Well, John, you've got some slides to go through.
Yeah, I've just got a few to try and break down some of those myths I was talking about before, and then
great.
We're going to have a bit more of a chat.
Thanks.
So I said earlier that one of the things that economists tend to do is only look at the costs of older people, not the contributions that they make. So I'm just going to have a look at some of those contributions in more detail, and then I'm going to look at some of the macroeconomic trends, which also run counter to many of the arguments that you generally hear. So in terms of the contributions, we spoke about this before. Older people, many of them are still in the workforce. I'm in the workforce, folks, and I'm not a burden on society, but I am an older person, no matter how you want to describe that term. Older people, are not just care recipients. Almost a third of older people are actually care providers. And yet, we tend to have in our mind this idea of a frail older person, burden on society, requires care and support. But in fact, probably the biggest caregivers in society are older people. And certainly the biggest volunteers in society are older people. These are all unquantified contributions which we somehow need to factor in when we're thinking about the balance between the costs and the benefits of older populations. As I said before, older people are still innovative. The most successful entrepreneurs are over 50. Older people still contribute taxes. And if you look at cash transfers within families, cash generally flows from older people to younger people until older people are well into their 80s. So all of these, and then older people make all sorts of cultural contributions, as you can see with many of the cultural icons who are still around, and people go and attend their concerts and things like that. And then all of that has been quantified, and it's been estimated that 7% of gross domestic product is provided by older people, and that quantifies in the US to $20,000 a year and 11,000 pounds per year. It says dollars, it should be pounds in the UK. So the older people are not just a cost, they also provide. if you look at the taxes that older people pay, the green and the blue lines relate to the green line. Sorry, this is age at the bottom, and the green line looks at the income tax that people pay. And because older people tend to move out of the formal workforce, you see that starts to fall around the age of 65. But the red and the blue lines, this is taxes on property. And not surprisingly, older people pay more taxes on property than younger people. And this is goods and services taxes. And because older people continue to consume, they continue to pay more taxes. And then overall, that comes to 35% of income tax in the United States is paid by people over 55 and 15% by people over 65. So again, older people make real financial contributions to society, which we often underestimate. This is a figure about how that impacts in terms of gross collections for two countries. On the left, the United States, and on the right, Japan. And what you see here is over the next 40 years, that the increase, the income generated from all forms of taxes will increase. The one that drops, and one that we really need to think about better, is contributions to social security. Because as people age, they tend to stop paying into social security, and so the income that's generated from that tends to fall at the same time as the population ages. So there is a real issue there that we need to think about. But in terms of tax income, it's going to continue to grow regardless of population aging. And then if you look at how all that adds up, it's amazing how little research there is on this. When I was at WHO, we tried to convene the top economists from around the World Bank, International Monetary Fund, and so on, to discuss how we might think about this differently than it's been done in the past. And it was very hard to get them to even think about quantifying those contributions that I just talked about. This is the only study that has really been done. It was done in the United Kingdom, and it looked at in 2010, total expenditure on older people was £136 billion.
I saw you earlier.
But when you look at the contributions that they make, it actually exceeded that by £39 billion. So the balance was very clearly in favour of the contributions of older people. And yet we continue to hear these negative stereotypes, which are basically a political agenda. People have taken older people as an excuse to push their political agenda for smaller government. And it's not a valid argument. They can push that argument if they want, but they shouldn't be using older people as the excuse. And then macroeconomically, what does that all mean? This is looking at the proportion of the population over 65 in 6 different countries, and unfortunately I couldn't get the colours to be the same on each side of the slope. But this is looking from 2000 to 2020 basically, and this is looking at per person GDP at purchase parity pricing over the same period. What do you see here? First of all, this blue line is the United States, because one of the places where we hear the most noise about the burden that older people are placing on society comes from the United States. This is Germany. So even here, the United States hasn't got anywhere near the proportion of older people that Germany has already achieved, and it never will. And yet on this side, if you look here, the blue line again is Germany. If you look here, this is the United States. You look at the growth of GDP per person over that same period, there's no difference. So this, and at the same time, Germany's put in place universal health coverage, it's put in place a universal long-term care insurance scheme, and it has a pension for everybody. These are things which the United States has not really been able to achieve. So don't tell me that population aging and putting in place effective systems to deal with it is going to hurt our society. It's not economically. And then another myth that really needs to be busted is the idea that population aging is going to place unsustainable pressure on healthcare systems. This is old data, but basically it looks at the contribution of different components to the cost of increasing cost of healthcare between 1940 and 1990, which was a period which saw more rapid population aging than we're currently experiencing. The estimate was the impact of population aging on those increasing costs in the United States was 2%. The real costs comes from the total inefficiency of the US system, new technologies which came in place, and okay, those technologies, they're used for conditions which are prevalent in older age, but basically the population aging was not the thing that drove the increase in costs. And then finally, when you think about health care, the biggest demands on the healthcare system aren't necessarily linked with population aging, they're linked to the proportion of people in the last 18 months of life, because that's the period which is most costly in terms of healthcare delivery. And if you look at population shifts from the perspective of the proportion of the population in the last period of life, here I'm going to show you the last 15 years, it's a very different picture than what you see if you look at the proportion of older people in the population. So for example, this dotted line here is what's called the old age dependency ratio. This is what economists fall back on all the time. And what it looks at is the proportion of older people against the proportion of people between the age of 20 and 60 or 65, which are assumed to be productive. So you're comparing the burden in their stereotype to productive components of the population. And you see this increase, and from that increase they infer this is going to place all these burdens, which I spoke about, and we need to cut services to avoid these unsustainable pressures. But if you look at the proportion of older people in age groups with 15 years of life expectancy remaining, it's this. And over the same period that the population is aging, the proportion of population who are in that last 15 years of their lives is actually falling. And so the pressure on healthcare systems is falling from that perspective. Obviously there is an increase in terms of demand from some of the chronic diseases which go on for longer than 15 years, but this is a very different picture. This is much more manageable, you know, and it's very different than the scenario we're often played. The next thing I'll just try and touch on, and then we can go back and have some questions, I think, is the idea of health span. And as I said when we were having our earlier discussions, often what I seem to hear here is we're not talking about health span at all, we're talking about disease-free span. And that's a really counterproductive way, I think, for Geroscience to look at health. And it's counterproductive for a number of ways. First of all, the reason that people are living longer these days is because they're surviving chronic diseases. So people who otherwise in the past may have had heart attacks are surviving those heart attacks, but then they continue to live with the condition. It's managed, their health in terms of their ability to do things might be pretty good, but they have the disease. When you then look at health span, you see people with disease, but you actually don't know what that means because we don't measure the impact of those conditions. So most of the things that I've seen in a number of the presentations here looking at healthy life expectancy or health-adjusted life years, these things are calculated by looking at disease prevalence. They're not actually calculated by looking directly at people's functioning. And one of the things which we've been doing, and I've heard also mentioned a few times around in some of the sessions I've been in, is we've been trying to take a functional perspective on looking at how you would define health and thinking about your ability to be and do the things that you value, and particularly the individual level attributes of that which are intrinsic capacity. And we can talk about that a bit later if you want to, but I've got a few more, or we can have some more questions.
Well, shall we move into the next phase then, John? So I guess in terms of what you're saying there, all of these slides are demonstrating that it's not so bad, right? I mean, in many respects, the data is very positive from the existing infrastructure that's out there. And when we consider much of the science that's been discussed today, some of the drugs that are being developed that are going to be dealing with those frailty issues that are at phase 2 clinical trial stage, maybe moving into phase 3. We've seen some of those pharma acquisitions starting to happen, or those licensing deals. How do we start to communicate the science of what's being discussed here at an event like this to policymakers for them to start factoring in the nuance of all of these technologies and the implications that they've got for these populations that we're talking about here? Because this whole industry is banking on the fact that governments are going to pay for these therapeutics and pharmaceutical companies are going to make them.
Okay, I think, first of all, yes, it's not so bad, but also we can be optimistic in the sense that if we invest, we can make it better. I think the thing about the bad view is it's like the sense of helplessness, and all we can do is cut services.
Right.
But I think what we have to do is reframe the discussion so we understand that the investments we make in health will make those curves better than they currently are. So that's number one. When I was at WHO, I was there for 10 years, and it's really hard to actually convince decision makers to think in a different way.
And who are those decision makers?
It's government, and they're very much driven by the economists, and economists are stuck in these outdated frameworks and these outdated paradigms.
Right.
So I think that we have to work with economists, and it's great to see people like Andrew Scott, who's trying to do this, work within the economic sort of field to try and get them to understand we should be looking at this differently. And then I think there are ways you can link the economic benefits even to cellular interventions, and we're not really making those connections.
So I always remember the time when I heard about the first weight loss drug. And the UK government, the National Institute of Clinical Excellence, said they were going to approve these drugs. And I was shaving at that particular point, and I nearly dropped my razor because they said they were going to deploy it for millions of people. And of course, we've seen the phenomena that happened on the back of those. Now, a lot of the therapeutics that we're talking about here, and obviously people in the industry are working on, they're getting closer to being ready to go, from phase 2 to phase 3, which means that if they prove their safety and efficacy, they're going to be on their way. Do you think governments are ready for this yet?
No, and actually it works both ways too, because the GLP-1s may have much broader impacts than just obesity.
Of course,
they may have more direct impacts on the entire aging process. So, it may be that there's already things out there in the market which will have broader applications, which actually becomes a more immediate cost to governments. And then you have the things we're talking about in the sessions here, which will come through later, but will also have similar very, very broad implications for society. And there are implications in terms of the cost for government, but there's also the implications in terms of the benefit for society, both for us as individuals, and then the contributions which we make together, which I would expect would counterbalance the direct costs. But again, I suspect what will happen is that when economists look at these things, they will be looking much more in the first instance at the costs rather than the benefits that accrue in terms of economic impact. So we need to really feed into that discussion.
Yeah, I saw a report from Swiss Re, which obviously is the reinsurer for insurance companies, and they were saying that these therapeutics are 35 years away, which to me is not real. They're going to be, everybody says 5 to 10 years, right? But they're going to be within 10 years, so we're going to possibly see them sooner. So how as an industry, we're here, this is definitely a step up, this event, right? So this is getting the political dialogue going, it's getting the big business dialogue going, and this is the first one, but there's a lot of echo chamber work going on in the industry now. So how, as a professional that's worked with the World Health Organization and other big organizations, how do we get this on people's agenda? How do we break out?
We have to make a really focused effort, because it won't happen by chance. And I think you're right. This is an echo chamber. I mean, literally it's an echo chamber.
Right,
But it's, and I've experienced this for 15 years. We've broadened it a little bit, bringing together the biologists and bringing together the people more oriented around social impacts of aging, but we're still just talking to ourselves. So we actually have to actively reach out and start to talk to these other people. And it's not our comfort zone. You know, it's not a comfort zone for somebody working on DNA methylation to go and talk to economists at the World Bank or, you know, International Monetary Fund. But somehow we've got to find a mechanism to make those bridges.
Yeah, I mean, I'm minded when you come to a conference like this of what happens in the COP events in relation to what's happening in environmental and climate tech. And obviously, the negative view is the fact that obviously the world is getting hotter, and obviously there's a crisis to be dealt with. And you've educated me today. I mean, I've always looked at the demographics in a completely different way to what I've seen today, which is, again, and I'm an industry observer, so, you know, obviously I need to do a bit more research. How can we get the politicians, the scientists, the policymakers, the investors, the pharma companies, how can we get them together? I mean, it is a COP effectively, isn't it, that's required?
I think there is an entry point. I think people are waking up to this issue that, I mean, we've heard for 20 years that populations are aging, it's going to place unsustainable pressures, and we need to deal with it. But I think people are starting to understand that we can intervene to actually make that a positive rather than a negative. And I mean people within government, people within these decision-making roles. So I think they're just starting to get receptive. But it will require significant effort and investment, you know, and the trouble is that each of us who work in universities, who work in our research centre, or, you know, we don't have the capacity to make that leap. I mean, we can't walk in to the Reserve Bank of the UK and say to them, listen, you need to be aware of this. So we have to create the mechanisms to do that, and that will require a concerted effort. It will require investment and the building of the skills amongst this echo chamber of a group of people who can actually form that bridge.
And we heard Mahmood talking yesterday about obviously the fact that we're in the Middle East, which is the middle of East and West. Do you feel that, you know, this is a place where it can actually happen? Because obviously the GCC is very active. There's a lot of capital. There's a very positive population pyramid. They don't want to, you know, have the same issues that we've perhaps in the West have had different societal implications of aging populations. I mean, do you feel this is where it could originate?
I think there's places like this where it really could change. We've got the evolutionary scientists in the audience there, and probably he'll correct me, but my take on evolution is if you have a very big population, you don't actually see dramatic change. What dramatic change evolves from is a segment, a small portion, or a portion of the population that's different, that actually changes occur within. And my experience in working with governments around the world is economies like here in Saudi Arabia, where there's rapid growth, there's a lot of thinking for the future and trying to think outside the established and outdated paradigms. Yeah, the potential is for a place like this to really make some of those changes. And it only takes 1 or 2 countries to do that successfully, and then suddenly everybody wants to jump on board. So yeah, I see a lot of potential here in Saudi and similar countries.
Well, that's great. And in terms of the policy implications at a government level, and we all talk about behavioural change, and I always think of French train drivers, you know, who effectively retire at 50, you know. How can we start to get people thinking differently about that sort of, you know, that the individual autonomy? How can they actually, how can governments affect those policies to encourage people to say, okay, well actually, yeah, you're fixated on a retirement age, that's probably something that you don't really want to start.
Well, we have all had embedded in ourselves this outdated stereotype of what a life is. You know, that you study till you're 25, work till you're 60, 65, and then retire.
Yeah.
And it's amazing how rigid that is.
Yes.
On top of that, we have these embedded stereotypes of what an older person is. You know, and that probably goes back to when we were children and we saw our own grandparents. That's what an older person is to us. Well, you know, I can remember. So the reason I left WHO was I hit their mandatory retirement age. So I had to leave.
You had to go.
I mean, talk about a stereotype of ageism, it's institutional ageism. So I had to leave, and then I'm lucky enough, because I've got transferable skills, that, you know, I could take up this fantastic gig at Columbia Aging Centre. But I'm the lucky one. You know, most people in society don't have that flexibility.
Yeah.
Most people, you know, so we need to break down those barriers. But also, I found there were barriers in my own mind because you internalize those stereotypes. And I was saying to myself, should I really be doing this at my age, And just to give some context, I've currently got a 10-year-old son. He was younger when I took up the job. Why would I think I'm any different than I was before? I'm relatively healthy. I can come skiing with me and try and keep up, So I think somehow we have to start shifting those perspectives inside ourselves. And one of the ways we do that is by modeling. And I hope one of the things by me continuing to work, other people will look to me. I certainly look to other people in our team. We've got an incredible 80-year-old academic on our team. I look to him for that inspiration. Actually that's the norm. The norm isn't to retire at 65. So I think the more and more people see alternatives, they might start thinking that, well, do I really just want to sit here for the rest of my life, or do I want to do other stuff?
Yeah.
But then we have to work on the other side from society to make it more flexible, to enable people to take up those new roles, and to break down the rigidities that basically keep them in that outdated stereotype.
Yeah, great. So we're going to maybe take some questions. So if anybody has any questions, that would be great. We've got a microphone to be able to run around. So just before we get the microphone to you, so John, one other question in relation to that. I call them the 35%. And these are people in society that are very demotivated from taking control of their own health span, getting off the sofa, getting off the pizzas or whatever it may be. How can we get to the point where, from a societal level, people feel included in this dialogue rather than excluded from it? I mean, it's a huge question, I know, but I mean, do you feel that there's a way in which we can break that cycle of people? I mean, we've passed peak obesity in the US now, which is not just the weight loss drugs, it's also gym membership, right? Diet management and so on. So you can see that these things can move the needle, but how do we actually go about doing that from an aging population perspective and ageism, as it were?
Yeah, that's a tough question, and because I think those behaviours are very deeply embedded by the time people get into older ages. But we also have to realise that our behaviours are not entirely of our own making. We live in an ecosystem, and our ecosystem probably encouraged and facilitated that those people ended up with those attitudes. So we need to build an ecosystem that's different and where people can see a future for themselves and see that there's possibilities beyond just sitting on the couch and doing those things. And I think helping them understand how they have control over their health, a lot of control over their health, is a starting point, but that's really challenging.
Well, it's an interesting sort of dystopian comment that in the UK, the health secretary, Wes Streeting, has said that he will make weight loss drugs available to unemployed people, which to me, when I first read that, was a little bit like, whoa, that's kind of, big government, all the rest of it. But the point is that I guess that they're trying to just nudge people and give people the facility maybe to break out of the cycle that they're in. So I thought it was quite a brave thing to do. We've got a new government, which is obviously left-leaning. So we have our first question. Thank you.
Well, thank you very much. Very inspiring and insightful. By the way, I would challenge you on the skiing part. So, but I'm a biomedical engineer, so maybe my economy is wrong, but I'm also an investor, and I see this prevention or predictive health innovators dilemma as not having a business model. Surprise, surprise, right? Now, looking at your statements, that particularly the GCC countries, for example, are much more forward and future-looking. It's a very critical question. Are we having a problem with democracy, that we're only thinking in terms of 4-year efforts, and that we are actually only doing things to maintain what we have? In Germany, we have a election coming up now too, and it's hopefully a changer. But I'm already skeptical that they think any way beyond the 4-year horizon to the next election. So what do we have to do to get a much more future-oriented look into the politics? It's basically the question.
Wow, I mean that's I totally agree. I mean I think these 4, in some countries 3 or even less year cycles are really debilitating, and they prevent us trying new things because they might have longer horizons. And I hadn't thought of it until you said it, but maybe that is an advantage that countries like Saudi have because they don't have that same cycle. Now, on the other hand, I believe in democracy, but I think maybe we need to have some sort of new system, but I think that's beyond the scope of this talk. But I'd like to take you back to your comment on why you don't think there's a business model. And also, we talk about prevention. Again, as I said before, I think one of the problems when we talk about Healthspan is we're very disease focused. When we say the word prevention, essentially we're talking about disease, preventing disease. I think we should be broadening it to preventing declines, and those declines may be much more proximal and immediate to people, and we may be able to convince them to actually take action. But I would have thought there's a clear business model there. If you can offer somebody a product which has demonstrated benefits and you can tell them that's going to slow your decline over the next 5 or 10 years, well, I'd take it. So it might be a small market segment, but I'd be surprised that it's not a business model. Okay. Yeah.
This is, again, it's very good, and it's not a follow-up question, but it's, if you ask an investor, he basically wants to de-risk. And one of the de-risking strategies or questions that they ask is, so how do we make money next year and the year?
Yeah.
And if you only limit it to a very low percentage of the population which is rich and affluent, you will not make it. And if you take things away from existing business, so you basically shift money from the sick care to a health span model, you will get a lot of, excuse my language, from a lot of people. And investors don't like that necessarily, but again, I'm just thinking we need to actually probably be a little bit more bold in that aspect.
Let me give you an idea about how we could really be transformative. So I said before that I view health from this functional perspective, from people's intrinsic capacity. If we were able to measure that in an agreed way, and there's a clear outcome that we all agree is meaningful to people, and then we funded health systems in terms of the impact they had on that outcome, which is clearly linked to a meaningful endpoint, and so could be FDA approved or whatever processes need to happen. Imagine how the health system would evolve, because the incentives would then be in place to actually deliver health, not to deliver throughput. So there's simple things which we can do if we can reach agreement on an endpoint like that and then embed it in the system, it can be transformative in the impacts. And I don't think we should shy away from that. I think we should be being ambitious and saying things are pretty stuffed up at the moment. We need to do it differently, and we've got a different model. And maybe a country like this, they can put it in place, and then people think, wow, that actually works.
Great. Well, any other questions from the audience at all? Thank you.
First off, I think this gentleman answered the question I was going to ask. I mean, it's really interesting to think about, right, in terms of democracy and longevity and all this. But John, you're an eloquent speaker. You communicate yourself well. Why are you looked as a leper on these governments and these economists? What's the pushback? What are these old paradigms that they come and say, John, I don't know about that?
I just think humans don't like change, and we were, all of us were brought up to think in a certain way. I'm a disruptor, so there must be something wired in my head a little bit differently. But for most people, they were trained and they're comfortable with that. And it's really uncomfortable to think outside that box, and it's really hard to imagine outside that box. And I think what you need, and it's really hard to explain to somebody who's inside the box that it could be different. They need to see it in action. So what we need to do is find a place or places which are willing to take some risks, which are willing to really push the envelope and try some new things. And coming back to maybe it's here in Saudi, maybe it's somewhere else. But what I get is when people see it happening somewhere else, then they can start to make that mental transition. Otherwise, it's really hard.
So John, we've seen certain countries around the world about, like, we saw the stats that you showed us with Japan. Obviously, we've seen the work that's been happening in Singapore, for example, which obviously is a much smaller country. A lot of tight integration between the NUS, the National University of Singapore. They've got a whole gerontology department, which is obviously now implementing national change. We've seen the same, in early steps in Israel as well. Are there any other experiences you've seen where actually governments are starting to look at this as a policy solution, which is bedding into the healthcare system?
I'll just expand a little bit on Singapore, because I work with the university and the government on a project called the Queenstown Project.
Right.
And it's actually trying to look at how you can transform the whole ecosystem to support better aging. And so they've taken this community of 200,000 people. They're rebuilding the entire infrastructure, including housing infrastructure, the way the shopping is designed, the way that precincts are connected, and also how health systems are embedded at a primary care level to sort of draw from those populations and enable them to maintain their health in innovative ways.
Yeah.
So again, it's these small places that sometimes are willing to take the next step and try something, and it's those sorts of pilots which I think will, because they're going to assess the impact of that, and if they can demonstrate that it has, positive impacts both in terms of people's health, their participation and economics, then maybe others will start to listen. There are a lot of initiatives. There's one initiative which I work with that I mentioned yesterday in the talk called Older People's Associations, which is entirely the other end of the spectrum. So we're talking about low and middle income countries across Asia and in Africa. And in those countries, they bring together older people within these communities. It might be a rural community or an urban community, and they try and help them assess their health. So they have regular health assessments. They try and put in place health promotion initiatives. They help try and build financial security by giving them microcredits to invest in things. And I visited one place in Vietnam where this little old lady who didn't have any financial security got a microcredit. She bought some chickens, and then she starts this small chicken farm which enables her to make a profit, pay back the loan, because they all have to pay back the money.
Okay.
So there are all these really innovative approaches which are happening even in poor places, which maybe we could learn about in the West.
Yeah, great. We've got another question from over here, please. Thank you.
Thank you so much for this informative conversation. This is Rana Al-Sabr from the Saudi Health Council, and I just want to know from your perspective, if I would ask, please list 3 different or main priorities for policymakers and governments agencies to start with addressing healthy aging, what would be? Because we're tackling health services, but not in specifically health and age. We're only focusing on the services and how we're improving the citizen health, but not specifically this topic.
Okay, well, I'd start by learning from the mistakes that all these other countries have made. Learn from the mistakes of the United States, which, doesn't even have a healthcare system. It's got a very funder-driven, provider-driven, fee-for-service system, which basically rewards throughput, doesn't reward health. And those systems also are very much designed around hospitals and institutional settings rather than primary care, and they're very much focused on reacting to disease presentations rather than preventing. So if you wanted the 3 things, I would look at how you can really build an effective primary care system, how you can make that system focus on prevention. And that's not just the doctor telling somebody not to smoke. That's putting in place comprehensive programs within communities which discourage smoking through, it might be taxation or making it hard to access, or having advertisements on packets showing people their health consequences. So comprehensive health promotion initiatives linked to primary care, And then within institutions, within hospitals, secondary and tertiary hospitals, trying to think about outcomes beyond just those related to one specific disease, and trying to look at the delivery of more integrated care. Because most of our hospitals evolved in era where it was acute conditions which were the main problem. And the hospitals are designed to identify an acute condition and cure it. That's not the world we're living in. You know, we're talking about chronic conditions today which require long-term management, and that requires much more integrated care that's not siloed. So trying to build institutions which break down those silos. So primary care, focusing on prevention and breaking down the silos.
Great, well I think that's a brilliant point for us to finish on, unless there are any other questions. No, great, well John, I've learned something today, so thank you so much for your time, it's been brilliant. Please.
biology obviously has a very central component in our day-to-day lives in modern life. I would tell you that that's true for our cells as well and for our bodies. So our mitochondria are organisms that are within our cells. The human body contains about 35 trillion mitochondria. So for each of us, we have hundreds and thousands of these organelles within our cells. You can see them depicted in yellow here around the nucleus of a cell, but they network more broadly throughout our organ systems. And so you can imagine these as a kind of cellular power grid. Mitochondria make 90% of the energy we need to survive. So literally, without mitochondria, there is no human life. They are what allows us to escape the second law of thermodynamics, which is entropy and decay. And so mitochondria literally keep the lights on for all of us as humans. As we grow older, our mitochondria start to become dysfunctional. And that can be driven by many things. Pollution, for example, unhealthy lifestyles, the aging process, and diseases of aging all contribute to mitochondrial dysfunction. And so these become highly relevant targets for diseases of aging as well as for the aging process itself. So what do mitochondria do? They convert the food we eat and the air we breathe into adenosine triphosphate, which is the cellular unit of energy, which feeds all of our cells. A typical adult is going to produce and consume your body weight in ATP daily. So that's about 70 kilograms of ATP daily. The organ systems that most require this are going to be our major organ systems. So the heart literally produces and consumes 15 times its weight in ATP daily. Our brain is producing and consuming 5 times its weight in ATP daily. And interestingly, much of that in the eye is actually used by the visual system, with the photoreceptors in the retina high consumers of mitochondrial ATP. Our kidneys are big producers and consumers of mitochondrial ATP, and our muscle actually uses about 45% of the ATP we use, and that's particularly noticeable during exercise. Our muscle have to gear up and produce energy on demand during exercise. What you're looking at on the left is really the accelerated aging of mitochondria. Those are old ones right there. This is how we start when we're young, so they tend to be elongated. The little lines you see in them is folds of the inner membrane, which is where the cellular machinery for making energy is found. And with age, we start to see these round mitochondria that really have big holes in that cellular machinery. With that, studies have shown that 95% of our muscle mitochondria are damaged with aging. And levels of mitochondria in muscle decline by about 8% per decade. So it's easy to imagine then, right, that mitochondria are ubiquitously involved across diseases of aging. And if you look around this circle, you're going to recognize some of those organ systems I mentioned. So sarcopenia or frailty, right, where our muscles waste as we get older, that's driven by mitochondrial dysfunction. Heart failure, whether it's with reduced or preserved ejection fraction, is a disease of mitochondrial dysfunction. Kidney disease, chronic kidney illness, which often follows from heart failure. Again, huge mitochondrial component to it. And diseases of neurodegeneration like Alzheimer's or Parkinson's are also mitochondrial diseases. We're going to be showing you data from clinical trials that we've done, particularly in diseases in ophthalmology, dry age-related macular degeneration, where the retina starts to decline over time. But I also want to point out that there are rare mitochondrial diseases that affect children. So these children have genetic defects which impact their mitochondria. And with that, from a clinical perspective, they are presenting with a form of accelerated aging. So some of what you'll have heard today is how hard it is to study aging because it takes a long time. We age gradually, starting from about the age of 30. When you look at these diseases in children, these rare diseases where aging is happening quite rapidly, it's still affecting multiple organ systems, it's a good microcosm, as it were, to try to understand how we might then turn to developing drugs for diseases of aging. With our drug, elamiprotide, which is in late-stage clinical development, we've demonstrated the ability to improve mitochondrial structure and function across a lot of different disease models. What I'm highlighting here to you on the left are 2 different disease models. So on the top, Dry age-related macular degeneration affects cells in our retina. It is the leading cause of blindness in the Western world in elderly patients. You can see in a healthy retinal cell that packed inner membrane, which is again the mitochondrial machinery to make energy. And in the middle, you see that obliteration of it during disease. Over to the right, with our compound, we can preserve that. In the bottom panel, I'm showing you animal models of an ultra-rare mitochondrial disease affecting children called Barth syndrome, again, presenting as accelerated aging, where you see that same trajectory in disease of obliteration of the mitochondrial machinery and restoration with our drug. What we've seen across models, and I should maybe move over here so you can actually see the screen, is that in old animals that are going through aging, experiencing declines in their heart, in their muscle function, we can see normalization of some of that by improving mitochondrial function. We've seen early clinical translation of these data in different diseases, which we'll talk about. So, Barth syndrome is one of the most severe mitochondrial diseases that I'm aware of. Children born with this disease have a 95% deficit in a key building block of the mitochondria, which is called cardiolipin. And that's what our drug actually targets. Because of this deficit, the disease can be embryonic lethal. Children affected by it, 85% of them die by the age of 5. And those who survive early childhood are going to present with a form of accelerated aging, where all of their organ systems start to compensate for that bioenergetic deficit. So the heart starts to remodel in a negative way to compensate for that lack of energy. Their muscles start to present signs of wasting and disuse, like sarcopenia. And patients who live long with this disease, again, don't typically make it past their 4th decade of life, but often can't go to school by themselves, they can't hold down a job, some of them can't even care for themselves, and that's in their 20s and 30s. We've done clinical trials in the setting of Barth syndrome, and our first new drug application is under review by the U.S. Food and Drug Administration for this disease. Decision date expected in April of this year, so coming up soon. The FDA did hold an advisory committee meeting, which is when they bring in experts to look at all the evidence and make a recommendation of whether the drug is efficacious and should be approved. We studied in this disease some of the older kids, age 12 and up, because it's very hard to catch the babies. They die very quickly. What we saw in older kids with this disease, who again, usually their hearts are remodeling to adjust for that bioenergetic deficit. Often they can't exercise well and they're declining over time. We saw improvements in how far kids could walk in 6 minutes of 100 meters, which is the length of an American football field. We saw improvements of 45% in their muscle strength and in parameters of their core strength, which the treating physician says means the difference between being able to go to the bathroom yourself or not, because you can get up. So improvements for these older kids in parameters important for their activities of daily living. And again, think of this more in the context of aging, where that's what we start to lose. There were also babies that we treated on a compassionate use basis. So these are children who are coming into emergency rooms around the world. We've literally had requests from Turkey, from Hong Kong, from India, from Brazil, Europe, and the United States. I'm going to play you a couple clips. One from the family's perspective of a child who was gravely ill, actually had to have a device implanted to support his heart function. It's called a Berlin heart, which was then explanted with our drug on board. And another from a doctor, Dr. El-Shakawi at Mount Sinai, who treated another baby, actually asked for the drug before the child was even born. And they'll tell you their experience. He testified before the U.S. Food and Drug Administration.
Declan is almost 2 years old, and in January, he was diagnosed with Barth syndrome. The extremely rare genetic disorder can cause heart issues, muscle weakness, and developmental delays. Declan's mom, Jamie Dubuque, says today her son is thriving. Declan came down with a cold, but it turned out to be much more serious, and he was transferred to the Children's Hospital of Philadelphia. Jamie says Declan was on life support for 2 weeks after going into cardiac arrest. He was gone for 20 minutes. Dr. Amy Goldstein asked the FDA for emergency access to elamipretide.
To date, he is the only child that needed that much support that did not require a heart transplant with Barth syndrome.
He's been taking the injection daily ever since. Jamie says now
he has so much
much energy, doesn't complain of any pain.
Okay, so one child from a family's perspective, this is another child from a doctor's perspective.
I'm a pediatric biochemical geneticist at Mount Sinai in New York. In January 2024, the NICU, metabolic genetics, and MFM teams all met with an expecting mother to discuss a worrisome and grim prognosis. At 33 weeks and 3 days gestation, this fetus was known to have a Barth syndrome with worsening cardiomyopathy. The baby was born at 36 weeks, 4 days gestation. We initiated ilimipritide a few days later. Initially, not much was different than the original prognosis. The baby's cardiovascular function was severely depressed, and early echocardiograms demonstrated an ejection fraction of 19% at 2 days of life. All the usual standards of care were continued along with ilimipritide, and for a time the baby required intubation and mechanical ventilation, vasopressor support with multiple agents, and the course was complicated with sustained ventricular tachycardia and arrhythmias. He was listed as status 1A for heart transplant. To put it mildly, this baby was very sick. By May 2024, however, the baby had demonstrated both clinical and radiographic improvements. Ejection fraction was now stable in the 40s. He was on room air. He was tolerating daily ilimipritide without any notable or apparent adverse effects. He was downgraded on the transplant list, and most importantly, he was able to go home. Since he was discharged from 24, he has continued ilimipritide at home. His most recent echocardiogram demonstrates ongoing improvements, and he is now delisted for transplant. To the best of my experience and knowledge as a triple board certified physician, I would not have expected this baby's trajectory to change as it has without ilimipritide. This baby has thrived against initial expectation.
So again, severe disease of mitochondrial dysfunction affecting children. They're born with a genetic defect, but the disease presentation and the course of presentation really mimics in many ways the process of aging. So this is relevant as we think about mitochondrial targeted therapeutics for aging patient populations. We are in phase 3 clinical development, which is the last stage of clinical development for ilimipritide, that same drug, for dry age-related macular degeneration. This is the leading cause of blindness in elderly patients. It affects over 150 million patients worldwide. It leads to significant lack of independence for older patients as they're unable to drive or do their activities of daily living or self-care. There are a couple of currently approved therapies targeting the later stages of this disease. They have not been associated with any improvement in visual function for patients. We found in a phase 2 clinical trial, 176 patients, double-blind, placebo-controlled, that our drug was significantly protecting photoreceptor health, which is declining in this disease. It was a 43% protective effect. It was highly statistically significant. What's really important, though, is that it was highly associated with improved visual function for patients, with a significant number of patients actually gaining 2 or more lines of vision. So to dive a little bit deeper into what we saw in that trial, and this is relevant in the context of age-related diseases, but also the aging process, the drug slowed the progression of photoreceptor death for all the patients we enrolled. But the patients who were most likely to improve in their vision were those with a slightly earlier stage of their disease and smaller lesions. And this is one of the big conundrums we face, right, in studying aging. You want to go into a trial when patients are rapidly progressing, but if you don't treat early enough, those cells might be too far gone to rescue and actually reverse. I'd like to talk about the potential of mitochondrial targeted therapeutics across aging, again, by illustrating what happens to various organ systems in diseases of aging and what we've seen in either animal or human studies, again, more broadly than the ones I've shown you in the eye and in rare diseases. So in the heart, as we grow older, our left ventricles start to compensate for our compromised mitochondrial function. They start to remodel, similar to what I said we saw in Barth syndrome. In Barth syndrome, we actually saw some reversal of that remodeling where the ventricles actually started to return to a more normal state. So intervening in heart failure, there is this potential, right? Instead of just reducing burden of disease by lowering heart rate or blood pressure, could we actually restore mitochondrial bioenergetics to the failing heart? In phase 2 clinical trials we did, which were short, 1 week to 1 month, we did see improvements in radioechographic parameters of cardiac function. We saw trends toward improvement in quality of life assessments or in functional assessments. And so again, this approach could be quite relevant to age-related cardiac dysfunction. We have certainly seen that in animal models. I talked about dry age-related macular degeneration, but for all of us as we age, whether or not we meet the diagnostic criteria for that disease, our photoreceptors start to die, and you get this compression of the layers of the retina. What we're seeing over to the right with both elamipretide and a pipeline compound that we're developing as an eye drop is really protection of those retinal structures in disease states. We've seen this both in the setting of dry AMD, we've seen it in the setting of glaucoma. A common consequence of heart failure in aging is kidney dysfunction. And so what you're seeing on the left is that the aging kidney starts to have reduced cortical perfusion or blood flow. It's indicated in red on that image. Over to the right, in a clinical trial conducted at Mayo Clinic in elderly patients coming in for PTRA, which is renal angioplasty, we saw that the placebo-treated patients on the left, where you're seeing a lot of yellow and red, really did have that impact on cortical perfusion, whereas we saw a protective effect with elamipretide and healthier blood flow, persisting 3 months post-intervention. And again, for all of us as we age, our muscle mass, strength, and function decreased by about 5% per decade after the age of 30, and even more terrifying for some of us in the room, about 10% per decade after the age of 60. So sarcopenia is one of the hallmark characteristics of aging. We did a clinical trial at the University of Washington in older patients with mitochondrial dysfunction who were otherwise healthy, but their muscles were starting to show signs of mitochondrial dysfunction. We can measure mitochondrial dysfunction in people by essentially, there's a technique called PNH radioplasty, where you can measure ATP production, and we looked at that in the thenar muscle group, which is between the thumb and the forefinger. We saw improvements in mitochondrial respiration that were significantly correlated with improvement in muscle function measured by force time interval in these older patients. Comparing this to what you would see in a similar group of patients after about 6 months of exercise therapy, it was comparable, and that was with 1 dose of drug. What about the brain? Our lead compound doesn't really get to the brain in therapeutic concentrations, so I'm going to talk a little bit about some preclinical data that we have with a pipeline compound, which was optimized for blood-brain barrier penetration. This is called bevamipretide. We've assessed it in models of ALS. We've looked at it in models of Parkinson's. We've looked at it in models of Huntington's disease, as well as frontotemporal dementia. Across these models, we see neuronal protection. These are data from one of the ALS models. What we see is reduced length and branching of neurons across diseases of neurodegeneration and a highly protective effect with a mitochondrial targeted therapeutic in this model. So in short, mitochondria might give us the path towards not just slowing, but potentially even reversing some characteristics of the aging process. We might be able to improve bioenergetics in aging mitochondria if we intervene early enough in the disease trajectory. For us, we are targeting a couple of diseases of mitochondrial dysfunction, late stage and dry AMD. We have some rare disease programs ongoing. But we do see broad potential beyond what we ourselves are studying with targeting mitochondria in disease, and you're seeing more and more progress in this field. Again, this is quite late-stage clinical development at this point. So as a takeaway for you, and I'll take questions after this, mitochondria are ubiquitously involved in human health and disease. We've learned a lot of lessons over, it's closer to 15 years of clinical development that we've been working on this. We have clinical data in multiple age-related indications, including dry AMD, where we're in phase 3 development. And we have late-stage development for some of our rare disease programs, which can inform some of this, and a deep pipeline. Our goal, right, is not just to keep the lights on, but maybe even to turn them back on for some patients who are undergoing the ravages of aging. So with that, happy to take any questions. No worries.
Thank you. Thank you for an interesting talk. So my name is Molly. I'm from KAUST in Saudi Arabia. My lab works on mitochondrial mutations. Mitochondrial mutations have been associated with aging and cancer.
Yep.
In your cohort, have you monitored the mutation load in the process?
So in preclinical studies, we've seen reduction of mtDNA mutations. That's in an aging model. So we have seen that preclinically. We haven't monitored it in our clinical trials to date, but in our phase 3 trial in dry AMD, we actually are monitoring that because that's an older patient cohort. I would say the limitation, right, is that if you're taking it from plasma, it's not necessarily going to speak to retinal mutation load, but these are 360 patient clinical trials, 2 of them that we're doing globally in older people. So it will be interesting, right, to inform whether that translates into patients. We've also seen that as a biomarker in Parkinson's, and we're doing work in Parkinson's as well.
Thank you.
Yep.
Thank you very much. Very interesting. My name is Fahad Al-Tuitri. I'm a cardiac surgeon at Triple Board. I'm very stunned with the results of the Barth syndrome and recovery with the cardiac remodeling. I wonder, have you commercialized or reached the commercialization stage of your product, or is it just based on funds so far?
So the Barth syndrome product is under review by the U.S. Food and Drug Administration, and so the first approval date could be April 29th, right? There was a positive recommendation from the advisory committee, so that usually translates to an approval. That would be our first step before commercialization.
Any idea about ischemic heart disease and mitochondrial bioenergetic recovery?
Yeah, so as you know, likely, post-ischemia, you see about a 30% reduction in cardiolipin, which is the target of our drug. In Barth syndrome, that's 70 to 95% reduced. In Barth syndrome, we saw improvements in that. In animal models, post-ischemia reperfusion, you see normalization, so you can protect against that 30% deficit. It's harder to do in a clinical trial setting because you have to make sure the patients are fully occluded and drug is on board ahead of time. But I would tell you that kidney trial we did, right, that's ischemia reperfusion for the kidney, and it was successful in that context. Yep. Any other questions? All right. Thank you, everyone. Thanks for joining.
I'm Jörg Bahler. I'm a geneticist, and I'm directing the Institute of Healthy Aging. There's a small research center at the University College London where we use different model organisms to study the basic biology of aging. And I will today talk about genes that are regulated and function in quiescent dormant cells. So processes, both the genetics and environmental factors which are important for aging, they're actually remarkably conserved from yeast all the way to human. And so you can use these simple model systems like yeast to study basic biological processes of aging. Yeast, for example, allowed us to discover autophagy or TORC1 signaling, clearly very important processes which are universally important for aging. But yeast is relatively simple, so that generates a useful basis then to understand more complex systems like ourselves. And so the aging model, a popular aging model in yeast is you have these proliferating cells when there's a lot of nutrients, and in the absence of some nutrients, especially nitrogen, they go into a non-dividing quiescent state. And how long they persist in this state before they die, that's called a chronological lifespan. That's a simple model for cellular aging. And quiescent cells, they are clearly important for aging and disease, if you think of stem cells, immune system, and so on. And they feature universal aging-related processes. And quiescent cells, they're very common. They're very common in our body. They're also very common for microbes out there in the wild, but they have been much less studied than proliferating cells. So in yeast, besides these quiescent cells, these stationary phase cells on top, which I talked about, there is also a even more dormant state, so they can undergo the cells of two mating types, they can fuse and they undergo meiotic differentiation. And they form these very, very dormant spores. And these spores, they can actually survive for hundreds of years if necessary. They have been revived from shipwrecks, for example. And so it's almost as if aging is suspended in this very dormant state. And then, so they can survive harsh conditions for extended time. And as soon as the environment becomes better, there's nutrients again, they germinate and they restart, the normal proliferation as vegetative yeast cells. And so this is still ongoing. We did a study of these spores, what happens in these spores. We looked at young and old spores, and also we stressed the spores to see whether these dormant cells can still sense the environment. And then we looked at the cells which germinated from these different types of spores. And when we took both RNA and DNA from all these samples to study the gene regulation in these cells. And so this is both changes in transcript levels and the protein levels. And you see there's a good correlation in generally. Genes, there are hundreds of genes upregulated or downregulated at both the RNA and the protein level. And you see some of the processes here. And an exception are, for example, the ribosomal proteins. They are upregulated at the RNA level, but actually downregulated at the protein level. So there's kind of an interesting regulatory anomaly there. And then we asked, does this gene expression in these spores further change if you age them or if you stress them? And actually both, old age in spores and stress generates a response. So they still change both the transcriptome and the proteome. And in a very similar way, whether it's being stress or old age, And you see some of the processes here. So that's quite interesting. That these are dormant things, but they still, reply to the environment and change as a function of age. And then we wondered whether cells which are germinated from these stressed or old spores, whether they kind of, have still some long-term effects on gene regulation. And that was quite surprising. So we germinated the spores, and then there are several generations, where they divide. But still, so cells derived from spores which have been stressed or aged, they show a different gene expression signature than cells from control spores. And that's, persists over several vegetative generations. You see some of the processes here upregulated. So there's like a memory effect. These cells, they remember from what type of spores they came from. And even more remarkable, that, that altered gene expression signature has also then cellular effects, like lifespan. So cells derived from spores, which were old, they have a longer lifespan. And the same is cells derived from spores which have been stressed. Actually, the more they have been stressed with a heat shock here, the longer the lifespan is. That's quite surprising. So you have this long-term effect based on what happened to them as a spore. We then wondered, does this yeast, so are some of these gene regulation effects conserved across organisms? And so we recently started to study an emerging model organism, the turquoise killifish. That sits kind of in a sweet spot here, in that it has a short lifespan of just a few months, similar to the lifespan of flies or worm or yeast, which have been kind of the workhorses of aging research. But it's a vertebrate, so of course it features more of the hallmarks of human aging. And this is quite a cool fish, because that fish that lives in Africa, in places where there's no water for most of the year, because they live in these temporal ponds, which only persist during the rainy season. And they survive the long dry season in a dormant stage, which is suspended animation. It's called embryonic diapause. And these embryos, they can dry out, and they can, really survive for a very, very long time in that state. It's almost as if aging is suspended in that state, because they can remain for 2 years or so. And, then when they hatch as a fish, they only live for a few months. So this is a kind of a unique system for invertebrate cells to study quiescent, dormant cells. So we used these fish embryos, and they did exactly the same, essentially, as we did for yeast spores. We looked at young and old embryos, and we also heat-shocked the embryos. And then we, again, extracted RNA and protein and looked at the changes in gene regulation. And similar as for spores, hundreds of genes are differentially regulated in these dormant embryos compared to proliferating, to actively developing embryos. We have similar processes. And again, the ribosomal proteins are kind of an anomaly in the sense that they are upregulated at the transcript level, but downregulated at the protein level. And then we ask the same question: does this gene regulation change if you look in older embryos or in embryos which have been stressed? And similar as we saw for spores, there is quite a strong additional regulation, and in a similar way, whether they are old or they have been stressed, that leads to a similar response at the gene expression level. And we also compared these data, so genes which are upregulated in spores and in diapause embryos, to genes upregulated in dormant cancer cells, another dormant state. And there is a fair number of genes which are regulated in common, either up or down. There's kind of a core set of genes which is like a signature for these dormant cells, which are universally regulated and conserved across organisms. That's both for induced and repressed genes. And you have some of the processes here for the genes which are upregulated. And the ribosomal proteins in all three systems, all three organisms, they are upregulated at the transcriptome level. So that's the conclusion of that part. Spores and these diapause embryos show substantial regulation of both transcripts and proteins in response to stress and old age. And in addition to stress and old age. And remarkably, cells seem to remember whether they encountered stress or old age as spores, and then results in additionally altered proteins in the offspring and longevity as well in the offspring. And we have this significant overlap amongst both processes and genes which are regulated in this different dormant state, very different state, yeast spores, killifish diapause, and dormant human cancer cells. And we have the cytoplasmic ribosomal protein show this differential regulation at the RNA and protein level, which suggests there might be some translation on demand. But also what was striking in these regulated genes is that many of the genes which are regulated in these states, they have completely unknown functions. Nobody has studied them. And this is something which really interests us now, and we want to focus a little bit. It's quite remarkable that researchers, most researchers focus most of their time on proteins which are already very well known. And actually, the more famous a protein is, the more it attracts further attention and funding, whereas others remain unjustifiably uncharacterized, hidden in plain sight. And this is really quite remarkable. This generates a huge gap in our knowledge, a blind spot of totally unexplored biology, which of course is a untapped opportunity for biomedical research. So you can see this effect, for example, in a huge bias in citations and publications. There are some superstar genes which, for example, for P53, there's more than 40,000 papers where P53 is in the title. And other, proteins are just completely not touched. And so, as I said, the more something is known about a gene, the more it attracts further papers. Just a few examples to highlight how much biology of this dark proteome awaits discovery. So we, recently, or in 2016, they published kind of a synthetic bacterium where they stripped off all the genes which are not required for proliferation, and they ended up with organisms of about 470 genes. So we kind of defined the bare essential for life, but remarkably, about a third of those genes had completely unknown cellular roles. From proteomic studies, about 40% of the proteins are not associated with any processes, including some of the most abundant proteins. And some genome-wide studies showed that about 44 of the most promising target proteins for Alzheimer's have never been appeared in any studies on Alzheimer's. And this doesn't, mean that these unstudied genes, they're not unstudied because they're less important. That has also been shown empirically. They are just essentially reflects historical bias and kind of the way science works, because biology is so complicated. You can focus all your attention and dig ever deeper in the same, old proteins. And it's actually quite hard, to get funding or to build a career on proteins where nobody, knows what they're doing. But I think you will probably agree that to fully understand, how cells work, we really need to know what all parts do. So even in very well-studied eukaryotes like yeasts, there's kind of a plateau, about 15% of the proteins. There's no real progress in the last few years. And we are involved in POM-based, that's a model organism database for fission yeast, and they kind of annotated a list of about 135 of these so-called priority unstudied proteins. They're conserved from fission yeast all the way to human across 1 billion years of evolution, but they have not been studied in any organism directly. Yet more than 50% of these genes are linked to complex diseases in databases. Diseases for which old age is the biggest risk factor. And now, given that these quiescent, non-dividing cells, that's an understudied state in yeast and everywhere else, it's perhaps plausible that many of these unstudied proteins might function in these quiescent cells in processes related to aging. And indeed, our recent data suggests that's the case. So I just give you a few examples. So we recently, globally looked at gene deletion mutants across many, many different conditions. And so you can make these phenotype correlation networks of mutants which have similar, phenotype profiles across the different conditions. And it's just an example. So you have these clusters of genes, and they always include this cluster includes mitochondrial genes and known aging genes, as well as 3 unknown genes. And so kind of this principle guilt by association, you would say, maybe these unknown genes also somehow function in mitochondria and aging, and that's what we find then if we look at them. So there's 1 short-lived and 2 long-lived mutants. They all affect lifespan. So we have a disproportionate number of unstudied genes that are upregulated in quiescent states. These stationary phase cells and the dormant spores, as I showed you. Also, many of the unstudied genes are, if you knock them out, they're either long or short-lived. For the long-lived ones, they are 3 times more than expected by chance, so there's a strong over-enrichment. This is done with, with genobite screens, barcode pooled screens. We find that more than 50% of the genes for which we see orthologs in killifish, they're up and down regulated in diapause. We get, again, an important quiescent, where we have quiescent cells. 45 of the unstudied genes, they are linked already with aging-associated phenotypes in databases, model organism databases, and GeneAge in at least 2 organisms or more. 10 have been linked with human GWAS studies. And there's this ongoing international mouse phenotyping consortium where 52 out of 66 orthologs, they are associated with traits relevant to aging or quiescence. So kind of all these intriguing links to aging of these unknown genes, they kind of set the stage to start to characterize them further. Yeah. Okay, so that's now in the last 10 minutes or so, I will talk about another set of unknown genes, and these are non-coding, long non-coding RNAs. And of course, there are many of those, right? Almost about 80% of our genome is transcribed, but less than 2% is coding. So there's a lot out there where we just don't know what it does, if anything. So it's clear that the transcriptome, the proteome in particular, is an important intermediate between the genotype and the phenotype. But it's not clear how these non-coding RNAs, which are actually more of those, how much they contribute to this information flow, and how they might affect cellular processes such as aging. And so this yeast, in fission yeast, we have many of these non-coding RNAs. They're typically very lowly expressed, but they shoot up, they become very highly upregulated in certain specialized conditions, which might indicate that they have specialized roles in these specialized conditions. For example, they're upregulated again in quiescent cells, in stationary phase cells and meiotic cells. So there's all non-dividing cells. And we did kind of genome-wide screens, looked at the mutants, deletion and overexpression mutants under many different conditions, and we find phenotypes. So in at least some conditions, for about 60% of the deletion mutants, we find that they're more stress sensitive or resistant, or the different nutrient conditions, they have a growth effect and so on. And even more so, if you overexpress them under many different conditions, we find phenotype for 90% of these non-coding RNAs. So they, at least seem to have some role. And now we focused on one particular non-coding RNA, which we called aging-associated long non-coding RNA, AL1, which we found boosts longevity. So this AL1 is strongly upregulated in these non-dividing cells, stationary phase, about 20-fold upregulated. And the question is, what does it do in those cells? If we knock it out, if you overexpress it, the cells live longer. If we knock it out, these are two independent deletions, the cells actually live shorter. So this AL1 seems to work as an RNA in trans and has an anti-aging effect. And we find then in yeast it's very easy to cross this non-coding RNA mutant against all the other coding RNA mutants, coding gene mutants. These are genetic interactions. So you then look for phenotypes which are stronger or weaker than expected, based on the phenotypes of the single genes. And that kind of puts you, allows you to put your gene of interest in a functional relationships with different processes. And if you do that with AL1, all the genes with which it genetically interacts have something to do with translation. We have cytoplasmic translation, ribosomes, all these processes. And this is, a strong indication that it somehow works in translation. And consistent with that, we actually find that the RNA, this is a single molecule fish, this RNA leaves the nucleus and is mainly localized in the cytoplasm in distinct dots. Most non-coding RNAs, they stay in the nucleus, they function in the nucleus, not AL1. And there's this method called chirp MS. You tag your non-coding RNA, pull it down, and you look with what RNA binding proteins it associates. And again, many of these proteins have to do with translation. And the targets actually form quite a big network, so many of the target proteins, they interact with each other, including a whole strong network of ribosomal proteins. And then, remarkably, so these are polysome profiles where you use a sucrose gradient and you separate the different ribosomal parts, polysomes and monosomes. And if you look at these profiles and you overexpress AL1, there's actually the content of ribosomes is lower in the cells. If you delete AL1, it's actually higher than in control cells. So it seems like AL1 can reduce the ribosome content, and that would, what may you expect that it reduces translation. We actually have some direct data that translation efficiency is reduced. And that would also make you expect if translation is reduced, cell growth of the cells should be reduced. So we tested that. Indeed, if you overexpress AL1, these cells, they grow slower. We then looked at, in AL1 overexpression strains, looked at the transcriptome. And again, what's really affected in this overexpression strain is mainly ribosomal proteins and other proteins involved in translation. Essentially, all the ribosomal proteins are downregulated in the overexpression cells. And so, it's not that clear how this could be. Is it somehow acting on transcription, or is it interacting with DNA? So we did again this chirp method. We have tagged AL1, we pull down whatever binds to it, which can be not only proteins, but also nucleic acids, and then we sequence. And we find that there's one messenger RNA encoding a ribosomal protein is very strongly associated with AL1. And this particular one is called RPL1901. That's in AL1 deletion mutants. This is higher expressed. If you overexpress AL1, it's lower expressed, about twofold. So it seems like AL1 inhibits the expression of this particular target gene. And if you use this Vienna RNA package, and you can kind of predict interaction between AL1 and RPL1901, they are actually strongly predicted to well interact to each other to form dimers. And I kind of poses the question, could this AL1 mediated inhibition of RPL1901, could that explain the lifespan phenotype of AL1 mutants? So we did different experiments. First of all, we just knocked out RPL1901, but that actually led to shorter lifespan. I mean, that's not surprising, right? You need ribosomes. And the AL1 interaction doesn't completely inhibit RPL1, it just reduces it about twofold, in fact. So we expressed RPL1901 with a regulator promoter to kind of tune its expression. And if we tune it just right, about twofold reduction in expression, then we actually see a longer lifespan. And that kind of suggests that this repression of RPL1901, this relatively subtle repression, that could actually explain the lifespan phenotype which we see at the cellular level. Remarkably, this non-coding RNA is not conserved at all. Most non-coding RNAs are really not conserved, unlike proteins. But if we overexpress it in flies, we actually see these flies live longer. So we overexpress the yeast RNA in flies, and that allows the flies to live longer. These are control cells. And it's not so easy to make flies live longer, right? It's very hard to make them live shorter. But so it kind of suggests that also the RNA itself is not conserved, but it interacts with conserved processes like the translation machinery. And so perhaps the principle, this anti-aging function of AL1 might be conserved. That's just the conclusions. I don't go through this. We can read. But this is the model of AL1 function. So in these non-proliferating quiescent cells, AL1 is upregulated. It directly binds to this one messenger RNA, RPL1901 for ribosomal protein, and inhibits it slightly. And then if this one is inhibited, probably with stoichiometric regulation, that's the idea that in everything, all the other ribosomal RNAs are also downregulated. Because they need to express at similar levels, right? Because if one is at lower level, you can't build a ribosome. So everything is downregulated, and that means then there's fewer ribosomes, less translation, and inhibition of translation has been shown before that this is good for cellular longevity. So we don't know all the mechanistic details, but that's kind of the model. How RNA has quite profound effect on cellular lifespan. And these are the folks and our funding, and I thank you. Questions? Dario.
Hey, you're great work. Just a brief question about the last part. So I maybe missed it. It's all the effect on lifespan that you observe in your AL1 mediated by RPL1901, or do you think there is a residual alternative activity? Is it, in other words, all kind of like linked to this interaction with RPL1901 RNA, or is something else?
We can't be for sure, but in a way, so this if we downregulate RPL1901 about twofold, that's what we see, what the interaction with AL1 has effect. That leads to a very similar lifespan effect as just a knockout of AL1 or the overexpression of AL1. So in a way, we wouldn't need anything else to explain AL1 function.
But have you tried? Well, I guess that's how you find it with a double mutant.
Yeah.
Okay.
Yeah.
Thank you. Great work. I just want to ask about the AAL1. Because overexpressing AAL1 is slow the cells, right?
It's slow.
It's slow the cells to grow, right?
Yes, yeah.
So have you ever done expressing this AAL1 in the cancer cells?
We didn't try that.
But do you think it's something that would slow the proliferation of the cancer cells?
Well, if that were true, that would of course be very striking. I mean, we were really surprised, with the fly result. We didn't expect that yeast RNA, which is not conserved, has any effect in flies. So, given that the translation machinery is so strongly conserved, and RNAs don't necessarily need to be conserved at the sequence level, to function, it would be really interesting to see what happens in cancer cells. Yeah.
But I'm worried also, just came to my mind, if you express it, does it will push the cancer cells to the senescence or senescence?
Sorry, I didn't quite.
Like if you suppress it in the cancer cells,
Yeah.
would push the cells to like senescence or senescence?
We don't know. Yeah, it's possible. Yeah.
From your experience working
We, in yeast it doesn't work like if you inhibit growth, it doesn't necessarily lead to senescence. But of course, if it would inhibit growth in consciousness, I don't know what would happen. It's a possibility, which would of course be very useful. Yeah. Okay, thanks again, folks.
both on interventions and biomarkers. So on the interventional side, we work predominantly on supplements and drugs, so small molecules that slow the aging process. And we realized as we developed these small molecules that we have to have ways to show their efficacy, to show that they actually impact human aging. And so to put that together, we need both the interventional side and the biomarker side. We need some kind of an effective biomarker to determine whether longevity interventions are having an impact or not. And I would argue that I'm not going to talk about interventions today. A lot of people talk about that at this meeting. We have our own. But this has been a revolution in research. There are now thousands of potential interventions that could slow the aging process. They work somewhere in animal models, or there may be some evidence in humans. None of them, I think, are fully validated. You could make an argument for exercise. But it's been really slowed down, and we need this coupled with it. We need to be able to measure aging, and we need to be able to use these biomarkers as endpoints in clinical studies. And I'm going to point out some of the challenges with that. And there have been many diagnostics of aging over the years. There have been functional parameters, physiologic measures, things like grip strength, pulse wave velocity, things you can measure. Those have value for sure. There's a number of clinical markers, like an individual inflammatory marker or HbA1c may correlate with aging at a very loose level, but it's not a really good predictor of the age. And then there are now aging clocks, and I'm going to spend a lot of time on that today. We can collect aging data from wearables. I have 2 of them on right now. There's questionnaire-based aging predictors out there. And of course, there are imaging, so full body MRI, things like that, that are measures of early stage or risk factor for disease. And all of these tell you something about the aging process. There are also composite scores, and I'm going to focus mostly on biologic age, but I thought I would just spend a couple slides defining what these different parameters are, because if you're an aging researcher, you might use one set of parameters. If you're a geriatrician, you might use another set of parameters. They're overlapping measures, and they're a little bit difficult to understand. So as an intro, I'm going to walk through that relatively slowly, and then I'm going to rapidly get into some of the challenges with the biologic aging markers. So, and I use perplexity for this, so we'll see how perplexity does, see what you think. So biologic age is a measure of how well an individual's body is functioning and aging at the cellular and molecular level regardless of chronologic age. So this is a measure of the age you're at biologically. It says regardless of chronologic age, but that's a little bit of a misnomer because many of the biologic aging clocks use the chronologic age of the individual in the algorithm to predict the biologic age. And so this could reflect the cumulative effects of genetic factors, lifestyle choices, environmental exposures, etc. So I'm going to come back and talk a lot about this. And there's been a tremendous amount of work on this, and I want to, Vadim's in the audience, he's been spearheading this, and I think it's trying to bring the whole aging field together in a way to validate biomarkers. And I'm not going to talk in too much detail about this. There's a couple other papers to follow this one. And I think it's great that the field is trying to come together because we need to understand what these different markers are actually mean and what they can be used for. I don't personally think there's a clear consensus on what the best clock is right now. Different people have different clocks. I'll tell you why we're using the one we're using in a minute. If you just search commercial biologic aging clocks, I just asked Perplexity to do this, it came up with 9. I don't really know how it did this. Most of these are methylation-based clocks, and I'm going to talk a little bit about that. This is glycan age, so this measures glycans in the blood. And then there's metabolomic clocks, and then there's 2 clocks that we helped develop, the clinical chemistry clocks. So the first thing to take home from this is you can use a lot of different parameters to try to measure age. Basically, any complex data set across a wide range of individuals can be used to predict biologic age one way or another. Now, the relative accuracy of those clocks may depend on the assays you're using. People are starting to use multi-omics clocks where they combine different kinds of things together. And so this is a rapidly changing field. Now, frailty is something that you hear about in a geriatric context a lot more. This is a clinical syndrome characterized by increased vulnerability to stressors due to age-related decline. And there are many different frailty scores. And I would argue that frailty, I want to come back to this question with biologic aging clocks in a minute, but I would argue that frailty, depending on how you measure it, some are physical measures, some involve social well-being, some involve economic well-being, but in general, they're very informative for populations. Whether they're valuable at the individual level is a different question. And again, what we're looking for here are, in this talk at least, are interventions how to validate interventions. And if things are not valuable at the individual level, there's too much variability, it's not accurate enough, then it's not going to be useful in that context. Intrinsic capacity is something the World Health Organization developed. It's a composite score of 5 different domains: cognition, locomotion, sensory scores, vitality, and psychological scores. There are lots of different ways to measure this, including questionnaires, and in some cases, functional measures. And again, I think that this is probably a more complete score than frailty, but again, will it work at the individual level? And another thing about these frailty, intrinsic capacity, is that they are disproportionately accurate in older populations. What we really need are some assays that work across the whole life table, not just after you're 70 years old. And so again, if you're looking for interventions, a lot of us are trying to do interventions in 40-year-old, 50-year-old, 60-year-old. We need scores that are accurate and don't have ceiling effects in that category. And then resilience, I think, may ultimately be the best way to think about aging, because it's a measure of the individual's ability to adapt, recover, and maintain or regain mental and physical health in the face of adversity. Adversity could be stress, it could be a virus. How do you respond to that? Do you recover? The challenge from resilience is that it's been very, very difficult to figure out accurate ways to measure resilience, because stress can come in many forms. And both in preclinical models and clinical models, I think people recognize the value of resilience, but it's been very difficult to measure. Okay, so this come back to biologic aging clocks. So the first two, you can use, just to catch everyone up to speed, you have a chronologic age and a biologic age. Chronologic age is your passport, and biologic age is what we're talking about measuring here. And you can use DNA methylation to measure those clocks, and I think that's a very informative way to go about things. You can also use transcriptomics, proteomics, metabolomics, any kind of omics. You can also use blood chemistry, which is something I'm going to talk about today. And you can use other things like activity accelerometry. So again, it's not specific to DNA methylation, although certainly the DNA methylation clocks are the most advanced at this point. And essentially what methylation's looking at are the changes, the modifications to your genome that happen. So different types of cells have different epigenetic patterns that's set up during development. And things are very ordered when you're young, but there are different kinds of changes that happen as you get older, including site-specific changes in response to things that happen as you age, and stochastic changes in the genome. And so you can measure this rate of change at a large number of sites and get an estimate of age from this. And these biologic aging clocks with methylation seem at least some level, I would argue at the population level, many of them work well. And if you look at this context, you can see the things that we know impact the aging process also impact biologic aging clocks. So in general, physical activity is associated with younger age, healthy diets associated with younger age, smoking and high-level alcohol use are associated with accelerated age, menopause accelerates aging, and a range of other things that we already suspected based on other kinds of data. And there are many types of clocks now, so I'm going to keep this simple. They're first-generation clocks, and these essentially were trying to predict the chronology or optimize an algorithm to the chronologic age of the individual, whereas the second-generation clocks are trying to predict an outcome, for instance, mortality, or some composite of diseases. And so these are really mortality predictors you can infer back to get a biologic age from. And then there are many other clocks that have come out, one of which I'll highlight is the Dunedin clock, which tries to measure the pace with which you're aging. So you can think of these other clocks as the odometer, which means how many miles you have on the car. You can think of the Dunedin clock as the speedometer, how fast you're aging at the moment. And I think this is an intriguing approach, and we use a number of these clocks in our academic testing. There are now organ-specific clocks based on clinical chemistry or methylation. And I just want to highlight that one thing that's not often lacking, but that's starting to change, is access to longitudinal data. Because a lot of these clocks, not all, but a lot of them are built on a cross-sectional data point. And even if you're trying to predict mortality down the road, you still only have one set of parameters to predict that from. As we get more and more longitudinal data, which measures the change of individual over time, I think it will lead to much more accurate clocks. I mentioned this. This is one of the first organ-specific aging clocks, a very simple one that I published with collaborators at Beijing Genomics Institute, and it tries to measure organ aging using clinical parameters. There are more accurate clocks that have come out from that now, and I think this highlights another question that we need to think about as we build clocks. What really matters when we're measuring aging? Is it a systemic measure of aging, which is one age for your whole body? Is it organ-specific aging, which is measuring how fast different organs are aging, or even tissue-specific or cell-specific? So I look at it as top down or bottom up. So if you measure all the different organs and then try to create a composite, that's more of a bottom up type of measuring aging. If you generate a system level component and then try to break down what the algorithms are actually using to make those predictions, you're starting at the top, and then in our case, when I'll show you later, trying to identify principal components that are the drivers of that prediction, and then trying to see what's in each component. So we tend to prefer top-down systems. Probably some combination of both is a great way to go. This is highly debated in the field still. But when we get to the individual level, there are challenges. And this reflects one of these challenges. This is a paper that came out, and it was 2 individuals that used 2 different clocks. I forget which 2 clocks they were. And they measured their biologic age like 9 times over the course of 80 days. And what you'll find here is that their variation is dramatic over this 80-day period, and actually the different kinds of clocks don't actually correlate that well with each other. So these clocks are not consistent as you measure them over time, and I think many of them aren't, and I think there's 2 reasons for that. One is that they're actually oscillating. So as you get sick, for instance, your biologic age might go up temporarily and then come back down as you recover. And if you decide to go on a health kick, you might get biologically younger, and if you stop, it might change. So there is a natural oscillation. Also, there's experimental oscillation. So recently, my colleague sent out blood samples to a whole range of different commercial DNA methylation aging tests, where he split the same sample into 2 different vials and sent them to various companies. And what he found was wildly divergent results. So there's also commercial issues now with replication of DNA methylation. And when you go to something like proteomics or metabolomics, it becomes even more of a challenge. I'm bringing this up not because I'm critical of these companies, but I think we have to go a lot further towards standardizing things if we're going to get accurate measures. And so when we're using aging biomarkers, we're thinking about how to do things. So we want to look at aging in populations. We have data coming out using 4 methylation clocks and 10,000 Singaporeans. We can see ethnic differences and make a lot of interesting inferences of biologic age. Just one of those I'll point out is that we were able to look at a birth cohort. So these are babies, they're just born, and we can already see a difference that the baby boys are older than the baby girls. So that's showing some very interesting data about development and how that may be already setting up differences between sexes in the aging process. But what we really want to use them for is endpoints and longevity interventions. And I think it's still an open question how meaningful that approach is. Another thing we really want is how do we do things that are actionable in clinical practice? I think there's been a challenge when someone goes to a doctor and says, these 10 methylation sites are predicting that I'm 10 years too old biologically. The doctor, in often cases, doesn't understand epigenetics. Even if they do, it's not actionable. They're like, well, what am I going to do about that? So that's why we built this clinical chemistry clock, and I'll tell you about it. We'd also like to predict disease progression. So I'm not going to show you this data, but my graduate student, who's now a postdoc, Max Unfried, built a lipid clock, a lipidomic-based aging clock, and he built it from pancreatic cancer patients. And to cut a long story short, we could actually, the biologic age of the individual at the diagnosis of pancreatic cancer was as good a predictor of their survival from the cancer as the best pancreatic cancer markers. So biologic age may predict disease-related outcomes and progression. That's one piece of data. We need to do this across a wider range. And of course, we want to use clocks to provide mechanistic insight. Can we work backwards from methylation or proteomics or metabolomics to understand more about how the aging process works? So there are many different uses for aging clocks, and some are more valuable in some contexts and some are more valuable in other contexts. So I want to spend a little bit of time telling you about our clinical chemistry clock, and then I'm going to talk a little bit about work from Peter Fedichev at a very simplistic level, but to try to give you some movies to think about what aging really looks like in a mathematical space. But first, let's start here. So, like I said, you can use all these different parameters, including even facial structure, to predict biologic age. But hospitals use this, clinical parameters, things you can measure in blood, medical histories. So we wondered, how can we build something like that that's usable? And so this is mainly work by Jan Gruber and Feng Sheng, who's a great young geriatrician. And we also wanted to predict something that predicts mortality, not just predicts chronologic age. So we had to use a historical data set. So we used the NHANES data set. So this uses demographic data, questionnaires, medical exams, and a lot of clinical laboratories. LDL, glucose, blood pressure. These are things that are measured in every hospital, in every doctor's office. There's tons of data sitting in hospital records, so if you could build a clock, you can work retroactively to look at what's going on. And I think one thing I've realized even more recently is there's standards for every one of these measurements. If you measure HbA1c here in the US and Singapore, you're going to get almost the same measure because these have been highly optimized clinical measures. So it removes some of the noise problem that we still have with methylation. Again, academically, we use methylation all the time. I think it's great. I'm just worried at the commercial level right now with the methylation. So we took this. There's multiple year input. Took 1 year to build the clock, and then measured outcome on the second year of input to see if it's accurate. And so you get something that looks like this, with this being chronologic age, and at the time we called it PC age. And you have people that look biologically younger by this clock and people that look biologically older. Again, this is predicting mortality, and then we're inferring backwards using demography to get to a biologic age. And so if you look at that in the next year's output, it works quite well. So we took people 65 to 75 in this group and looked at their survival here. So the black line is the survival of the whole population. This is thousands of individuals. The red line, sorry, too far. The red line are the ASCVD scores. So this is one way of predicting survival. It's using cardiovascular disease score. So it's looking at a system in the body that's causing the highest level of mortality and seeing what it predicts. So the 25% people that have the best ASCV scores survive better, and the lowest quartile have a higher mortality. So when we looked at our clock, we're better at predicting early mortality, but I think more importantly, we're way better at predicting the people that are going to survive 200 months out from the experiment. So it looked like the clock was working, so we looked at it in somewhat more detail. So you can map all the different principal components onto one graph just to give you an idea of what this looks like. And what you can see is there are green circles of people. These are groups of people that cluster together. And these are the people we predict are going to survive. And then you can move in this graph away from health. But you don't move only in one direction. There are multiple directions. And because we know what's in each principal component, we have an idea what people are moving in which direction. So this large group here, these people have cardiovascular disease or metabolic disease. There's a group of people going this way that have frailty and other kinds of disease together, but not metabolic disease. And then this is more specific diseases out here that cluster together. Now the thing we wanted to know is, since this is a principal component-based system, we can look at each component. Is there a component that tracks with distance from this zone, not direction? So it doesn't matter what you're getting, it's how far you are away from health. And we did find a component like that. And the list of things are here. And if you look at it, it's perhaps not surprising. It's basically immune system inflammation. And so this is reinforcing this idea that inflammatory events, changes in the immune system, may be principal drivers of aging. And then specific behavior and genetic risk may define what diseases and what paths you go down. So if you look at what's here, it's immune system here, red cells here, inflammation, relationship to kidney, and so iron storage. And so each component is different. And so when we get somebody that has a high biologic aging score, we can go back and find the component that's off. And then those are actionable components. Everything in each component, there's treatable outcomes in there with drugs that already exist. It seems to work. We generated a similar clock. I'll tell you a little bit how we did this. So the calorie study is a long-term study of calorie restriction, but they don't measure every last thing that's in the NHANES data set. There are a couple that are missing. So we took those parameters out of NHANES, went back and rebuilt the clock, which was almost the same, and then applied it to this study. And what you can see is that the people that are on the normal diet are getting older by 2 years roughly during the 2 years of the study, and at least by this clock, they're protected from changes in biologic age by calorie restriction. Some of the methylation clocks don't show this, others do. So at least this validates in a different study, and we're now validating across a wide range of studies. There are also some interesting things you can get out of this. You can start to look at trajectories of different individuals. We know what diseases people had, and we know they're indicators of each clinical component. So we know what medicines they're taking. So we can take this group of people here. These are people that are optimal or near optimal in every reference range. So everything that measures is in the normal range. And that goes down with aging. You can also see people here that are requiring treatment. So they have something out of the reference range, but they're not taking the appropriate medication. So for instance, they have high blood pressure, but they're not taking blood pressure medication. And that goes up with age. So these people are being poorly treated medically. And if you look at survival, they have a higher mortality. They were only looking at 120 months here. They have a higher mortality than the control group. If you look at the optimal group, they have a lower mortality. This is all predictable. We would have expected this. It's more interesting when you break it down. So first of all, if you look at the age-related principal component, you can see that the optimal people that are on no drugs have a lower biologic age, which is again expected. But what we didn't expect is that the people that are actually taking medicine that have optimal clinical chemistries, so these are people that at some point had high blood pressure or high glucose and they started taking metformin or beta blockers or something, but they look good now, they have a lower biologic age than the people that we're saying are okay and not giving them anything. And that shows up in the survival too. So the people that are taking what we call a polypill here, so they're medicated but optimal, are surviving better than the people that are taking no drugs. So the biologic aging clock corresponds to that, and I think that's really interesting what that might mean. One interpretation of that would be, there are multiple interpretations, some are less interesting. I'm only going to give you the interesting interpretations. One of them is that we're not being aggressive enough with people when they come into the doctor's office. So they're 60 to 70, they come in, these reference ranges are wide. We say, oh, for a 65-year-old, you look really good. Things aren't perfect, your blood pressure's 130, but just keep doing what you're doing great. And we should be more aggressive with those people. Now, maybe not with medication, maybe we should be more aggressive with exercise or diet. It doesn't have to be drugs, but we need to be more aggressive. I think the other thing that's interesting to me, though, is the idea that we've been treating aging for a long, long time, because these early risk factors for disease are basically markers of a network breaking down that's leading to aging. And so if you catch them early enough and treat them, high glucose, high blood pressure, high cholesterol, you not only prevent a movement down that particular disease trajectory, but you, to some extent, restore the network that keeps you functioning well, and that protects you across other things. So there are other studies that have similar results, and I think the conclusion here is that for whatever reason, the people on medication are doing better than the people off medication in this group of people. So it's interesting to think about. More recently, methylation data came out on the NHANES data set, and it allowed us to compare how our clock's doing to some of the other clocks. And it's, essentially we do better, or statistically better than most all of them, and better non-statistically significant than all of them. And so these are different clocks, and we're looking at how the age prediction of those correlates with mortality. And so the lineage 2 is the one we're using. That's here, it does better. This is what a perfect clock would do. This is better than all of the other clocks that you see here. Some of them do better than others. Some of the clocks don't predict survival better than the chronologic age of the individual. That's a bit scary. Other ones predict pretty well, like Grim age, but So you can start to compare how different clocks function. We're also very good at predicting cognitive score and gait speed using this clock. So we're pretty confident that we have something meaningful here, although we have to expand it to other ethnicities and races. It's still driven on an American data set. And finally, you can actually look at individuals here. So these are two different individuals, and these are the different principal components for that individual. And so the first individual has a high BMI, and they were a moderate smoker. This is, I think, taken at 72, and we predicted a high biologic age that they survived 5 years after this test. And then you can say which components are driving that, and in this case, I think this component is the one that's linked to smoking, and this component is basically metabolism. So if you go look at principal component 1 and break it down, it's mostly cardiovascular function and metabolism. So that makes sense. If you go to someone who was doing quite well, they're mostly blue in these parameters. They were 72. We predicted their biologic age lower. They died at 91. So the point is that you can actually go in and be clinically actionable here. You can say which component is driving this prediction, and then almost all of them are medically actionable components. And what we ended up seeing is that a lot of people are not out of the reference range, but if you combine 4 or 5 things together that the computer put in one component, it's driving their increased mortality risk. So maybe this is a way to get to early treatment. The other thing we've noticed trying to use it in clinical practice is that in about 10% of cases in older people, we catch something they didn't know they had. So one example is a person came in, looked fine, were being treated for everything, their biologic age was high, and so the doctor looked closer, Feng Sheng looked closer, and the person had a skin rash, and they'd been to the dermatologist, and the dermatologist said, oh, it's just a rash, put some cream on it. It turned out to be a skin infection that was accelerating couple of the parameters. Once that was treated, that came back down. Now, what does that mean? Is that aging or not aging? Well, these parameters are linked to mortality risk, so I think it's an interesting thing we're probably working at the interface here between aging and disease. Okay, so just to talk a little bit about some work by Peter Fedichev and Jan Gruber, because I think it gives you a good graphic representation of how aging might work mathematically. And Peter looks at very large data sets, UK Biobank and other huge data sets, and tries to use physics principles to extract what's going on with aging. And one of the things in his analysis that he often finds is that the first major principal component is one that tracks with aging. So if you look at this fly data, there's a difference here in one component by sex, but the second major component here predicting variability is age. And so he's been trying to look at that in more detail, and I'm probably not the perfect person to take you through this story. This one slide is really slow. I apologize for that. And so, in essence, if you think about things, physiology and feature space, you can compare any 2 parameters. And if you look at those parameters, it could be pretty much anything. I'm going too fast. All right, I'm hesitant to. I got to get Jan to give me a better slide here. It drives me crazy. Could be things like inflammation and cardiac function or catabolism. Doesn't matter what you look at. You can map it on a graph like this, and there's an optimal physiologic state for people to be in. And there's a range outside of that optimal physiologic state that most people fall within this range. And maybe this is a measure of resilience, but if you think about what's going on here, maybe, is that when people, all right, I'm officially going crazy. When people fall out of this optimal range, it's like a spring. Resilience sort of brings you back into that optimum when you're healthy. But when you go too far out of the range, you can go over an activation barrier and enter into a failure state. And so healthy people are almost all in these states, but once you go into these states, you have chronic diseases. And you can think of this in 3-dimensional space as the depth of these valleys in a graph. And so if you have healthy people, things are happening to these people. They're moving around, they're getting sick, they're exercising, they're doing all kinds of different things, and these parameters are moving. But since they're effectively deep in this valley, they keep being brought back toward the healthy state. So that maybe this is what resilience is. These hills are very big to get into these failure states. This could be cardiovascular disease or diabetes or anything you want it to be. But almost nobody's going there because the gravity is really pulling people back down. Now, it's interesting to think about that because when you measure accumulation of damage events, it's mostly linear. And I think what Peter would say is that he thinks of it as death by 1,000 different causes. There are a million small things that can happen to you as you get older, and they're relatively stochastic. And it could be DNA damage, it could be changes in methylation, it could be protein misfolding, it could be anything. And there's enough, all of these things are extremely low probability on their own, but they accumulate over time, linearly, in the measures he does. But as long as you have a network in place, it compensates for the damage that's occurring, and that's what's defining this gravity. Over time, what's happening is this network that's keeping you healthy, this homeostatic network, is breaking down. And the net effect of that is the size of the hills are going down. And so what that equates to is that, sorry, slow again. Yeah, they're going down. So what that equates to in the young state is that you have this resilience that's pulling people back to health. But in an older state, when you diverge off the optimal range, there's less to pull you back. And so it takes longer to get back to health. And sometimes you go over into these failure modes. And once you're in these failure modes, you're in a chronic disease state that is a different state entirely. And so I think this is how we think about aging. And what we're really trying to do is to maintain the height of these hills. And so if you treat a disease progression, you may block this trajectory, but there's 20 other failure states you can go down. And so you're treating a disease. If you maintain this depth, you're protecting against all of the failure states at the same time. And that's what we think longevity interventions are trying to do. Okay, and what it does is it provides an interesting thing because you can do the math and look at movement over this activation barrier. And what that means is that you go from a linear accumulation of damage to a Gompertz-like change in the failure state. And so you can map this onto a Gompertz equation, which is an exponential increase in mortality. In humans, mortality doubles about every 8 years. So it allows you to transform linearity of a damage accumulation into exponential increases in mortality. And you can think about this in the context of physics, and there are terms for this in different fields. So you can call it eigenvalues, you can call it intrinsic capacity. It really looks like resilience, right? And so what it does, no questions from me. All right, nobody's else asking questions, you can ask them. Yeah.
Thank you for a wonderful talk. I'm actually interested in finding the biomarkers in young generations. As a scientist in the stem cell research, we have recently observed that some of the bone marrow stromal stem cells that we get from young individuals, they exhibit high senescence and they won't grow.
Yeah.
So we go back and we report to the clinicians and then they investigate further. But we've been getting more and more of these cases on which we recruit these subjects as healthy subjects.
Yeah.
They turned out to be an actual patients.
Yeah.
So have you encountered this? Because this has been increasing.
Yeah, I mean, this is particularly relevant with things like diabetes, which are like super prevalent now, and young people under 10 years old. Type 2 diabetes is growing dramatically. So I think we need to think about this. A lot of these clocks were not made on including childhood populations. They're built from, say, 20 to 80 age range. And so they are less informative when you apply them outside of that age range. A few of them were built that way. So we took 4 clocks from that 10,000 Singaporean study. 2 of them were not built on young populations, and they were meaningless when we applied them to babies. The other 2 included young populations in the data set when the clock was built, and they were relatively accurate when we applied them to babies. So I think it's important to go back and build clocks that have that age range in it. But it certainly could be more testing done with some of the clocks on that now. I'll give you another example. You know, we built, in Haynes was people 20 to 80 years old, and then we tried applying it to people at a senior living home in China. And so they sent me the first data from a 95-year-old, and it said they were biologically 101. And I'm like, I'm not going to go tell a 95-year-old they're biologically 101. So I think it's important you have to use the right age range to build the clock. So that's a limitation, but I think it's a great question, and some of the clocks may be applicable there. So, yeah.
Thank you very much, Brian,
Sure.
for this
Anytime.
insightful presentation. Actually, I have two questions. One is actually that, we have different biological clocks nowadays. You mentioned actually a list of them. So, and also the literature is actually full of controversial argument. And some actually they're not in favor of, for example, the epigenetics and the methylation clocks, and other actually would favor like, for example, glycomics, also lipid clock and proteomics and others.
Yeah.
Would you please, when it comes actually to, for example, the reproducibility of the data and robustness and etc., which approach would you actually prefer? And if you give like some comments with regards to this, if you allow me, maybe I can take your answer and ask the second question.
Yeah. Well, I think the first thing is that we have to, there's longevity clinics everywhere. People are trying to apply this. I work with them. I think it's great. Having said that, this is still research, and so we're in this strange place where we're still doing advanced research on interventions in aging and measuring aging, and yet clinics are applying this at the same time. And I think it's fine, but we still have to realize that there aren't definitive answers to a lot of these questions. And I'm not sure I could give you what was the best clock, for instance. When we do academic studies, we measure many different clocks and look at changes in each. I suspect that when we think of it from an interventional point of view, we'll have a list of interventions here and a list of ways to measure diagnostics or clocks. Some interventions are going to work better for some clocks than others. And then when they turn back those interventions, that's going to have overlapping but disparate effects on the physiology of aging. And one of the things we haven't done well yet is we have all these verticals: interventions, clocks, hallmarks, outcomes. But we don't have ways to work across these verticals very well to predict what intervention is going to have impact on which of these other three verticals. And so this is still emerging research. And I think we're at this challenging point, because at one point we want to define rules for how biologic clocks are done, and I think that biomarkers are done, and that's great. On the other hand, we need to keep the freedom open to keep developing new and different strategies to build clocks, because I don't think we're at the final point yet. So again, we're somewhere along the path towards standard operating procedures, but we're not there yet, if that makes sense.
Thank you very much. Well, also continuation of this, this first question is, you mentioned that, and also this actually also shown actually in the body of the literature, that even if you measure the same samples, okay, and if you, for example, the same person actually exposed to certain stressors, for example, or after exercise or whatever, actually you can find like different readings for this. And for me, actually coming from the stem cell field, and in particular the pluripotent stem cells, and we know this cells, if they are in the culture, even though if they are all homogeneous population, if they are stem cell pluripotent, they are not exactly in the same core of the definition of the stemness state, if you want to call it.
For sure.
Okay, they will go like,
for sure,
they will like swing, in that field until they jump out of that state, and then they cannot go back. Is there like a threshold line that the methylation clock can tell us where are we in this?
People have tried to apply methylation to cells in culture, and so I'm not sure if I can directly answer that question. I would say, though, that you're bringing up another challenge, which is that if you take a sample from the body, like PBMCs or even saliva, you have multiple cell types in there doing multiple things. And so if you do a bulk assay, you're averaging or doing some composite of all those different cells. Whereas if you do, ideally you might want to do single cell, which would tell you more information, but that's not applicable at a commercial level right now. So, and we're not sure still, I think, what all of that means. So it's, the variability is inherent, but I don't know if it predicts the transition into a pluripotent state or not. It's an interesting question. Yeah.
Thanks a lot, Brian, for this great presentation. I actually saw the roller coaster in Copenhagen. It was very exciting already at the time.
Yeah.
As a biomedical engineer and not a biologist, I would like to ask, what is the chance that we'll actually find a meaningful digital biomarker? Because at the end of the day, this is an expensive process, right? And maybe also, it is an invasive process to a certain extent. So is there any chance we get rid of this blood drawing all the time, or at least less frequently? So we replace some of the things, some of the updates with digital markers?
Well, I think, let's talk about wearables for a sec. I think wearables are getting better. I mean, I'm pretty impressed with, I've just tried out this Whoop. It doesn't understand me very well because I travel on airplanes all the time, and I'm like the only person in the world apparently who sleeps better after they do airline travel. But that's an aside. I think that the wearables are getting much better, and they're starting to measure things like blood pressure and all kinds of other things. So I suspect that we're getting more and more meaningful aging data out of, I'm not an expert in this field, but just from listening to people and from personal experience, I feel like they're getting to the point where they're going to start to tell us really accurate things about aging. We have an expert, for instance, in Singapore who studies sleep, and he studies aura rings, and it's pretty amazing what they can infer about sleep and population levels again from the aura ring. Now, whether that can be applied at the individual level is the same challenge as all these other clocks. So, but I'm optimistic they're moving in the right direction, certainly. Yeah. Okay, I think we're out of time. So thanks for coming to the talk, and I'm happy to talk afterwards. So, thank you.